Good morning, everyone. I'd like to welcome you all today to our annual Department of Medicine Whiting School of Engineering Research Retreat. My name is Nicholas Zakos. I'm an associate professor in the Department of Medicine in the Division of Gastroenterology and Hepatology. Thank you all today for joining us in this very unusual virtual event. We hope everyone has made it through this very exciting and challenging year. We are very excited today to bring you a number of exciting and engaging talks today. We're really excited to be able to kind of host this format without too much disruption or changes as we've done in the past. And we hope that you guys will all find this to be a very engaging environment. Particularly with our poster presentations, which we're excited about to send you all into a virtual version of our own Turner Auditorium, as we've had in the past. I would like to begin today by briefly going over the agenda. And today we were going to start with the Spotlight on Research session. This is very much a highlight of the outstanding research and accomplishments that were achieved within this last year out of both the School of Medicine and the School of Engineering. This program was developed by Peter Abadir, a member of our research committee and former chair of our research committee, and along with Dr. Stephen Cohen, who will, both of them will serve as the moderators. And this year, we're excited to present four speakers from both the Department of Medicine as well from the School of Engineering. Again, highlighting some really outstanding achievements that they have made in the areas of research. Today, we are also excited to be able to welcome Dr. Taikchip Ha, who is one of our esteemed Bloomberg professors and a Howard Hughes investigator. And he is going to present his really amazing work in using and employing new technologies and for advances in CRISPR technology and how to really take CRISPR technology and move it along the way from the laboratory and eventually to reach the clinic. This will be followed today by a brief introduction from Allison Moliterno, who is associate professor in the Division of Hematology in the Department of Medicine. She will give an introduction onto how CRISPR technology is being used currently in clinical trials and in clinical practice. She will host a panel discussion today, which is going to be really an exciting panel. I was able to get a preview of it yesterday, and I think you guys are all in for really a treat today to hear a wonderful discussion about the pros, the cons, and the excitement within gene editing in terms of clinical applications. And of course, we're just so privileged to be able today to have a recent Nobel laureate in chemistry, Dr. Jennifer Doudna, who is going to talk with us today about obviously her groundbreaking work in CRISPR technology. And this keynote address today is going to serve as the Medical Ground Rounds presentation, and would like to send a special thanks to Dr. Sanjay Desai, who was very willing to allow us to move Medical Ground Rounds from its normal Friday time slot to today to accommodate this really unique opportunity to hear from Dr. Doudna. Tomorrow, we will have opening remarks, as we usually do, from Dr. Mark Anderson and Dr. Ed Schlesinger, who will give us an update on the achievements that have occurred within both schools over the last year. And then we will dive into this virtual platform that we're really looking forward to everybody participating in. We will have a 2-hour poster presentation gathering. It'll be a virtual setting, and where everyone will have the opportunity to almost kind of walk through a virtual world, and as you would in the Turner Auditorium, and go from poster to poster and be able to directly interact with each of our presenters, as well as those who are actually visiting each poster at the same time. You'll actually hopefully almost feel as if you're in a small group together. We will then be followed by, as usual, our research award winners. The announcements will be made tomorrow around lunchtime, and following that, we will have awards presentations given by the faculty members, both in the basic sciences, also in physician scientists, as well as the Outstanding Award, Laboratory Award from the Whiting School of Engineering. So as we've tried to encourage all of you in the past, I'd like to encourage you again, please, for those on social media, we ask that you please really get our message out there today. Please use this opportunity through Twitter, through your other social media platforms to inform, the scientific community at large about all of the great things that you hopefully will see over the course of the next 2 mornings, and really get the word out, and hopefully generate more excitement about not only what is being done here, but what we are highlighting moving forward. So I would like to take the opportunity to, of course, thank all of the members of the Research Retreat Committee, particularly Dr. Claire Kerr, who is the co-chair along with me for the Department of Medicine Research Retreat. We have the privilege of working with an outstanding group of colleagues who have done made an enormous sacrifice over the last year in terms of adding more Zoom meetings and putting in a lot of time and effort and creativity, and hopefully putting together this really outstanding agenda for our Department of Medicine retreat this year. She is joined by a number of other colleagues here in the Whiting School of Engineering. I thank all of them for their work, and also really want to thank also our colleagues here within the Department of Medicine, who we have a number of people here who have served as former chairs in the past, but who have just enjoyed this process so much they've stayed on and really helped keep this high quality research retreat moving forward, and also adding new things every year. So I very much appreciate all the efforts from all the members of the Research Retreat Committee, both from the School of Engineering and the Department of Medicine. So I thank you all. And also a very special thanks to our administrative team. We have had just an outstanding group of people who have helped put this event together. This is the first time we've obviously ever had to do something along these lines in terms of a virtual event, which I dare say I can't wait till we get back to in person. I think it should be a much easier event to put together, hopefully next year once hopefully we're out of this. But again, a very special thanks to everyone, Kelsey, Helen, and Holly, with our reps from the Department of Medicine, and Emily, Melanie, and Alexander from the Whiting School of Engineering, who've not only just organized this, helped with the communications, helped us putting together all these links, and also setting up our virtual platform for the poster presentations tomorrow. I can't thank you all enough for your help with all of this. And also thanks to our production team, Hiro Amano and Gus Sentimentes, who are basically our studio team, who are helping put this production in place, guiding all of us on how to do this in the right way. So I thank everybody for, again, their assistance with this. And I will give a special thanks very briefly to Dr. Stephen Cohen, who is going to be introduced officially and more formally by Dr. Peter Abadir. The two of them are going to moderate the Spotlight on Speaker session in the next few moments. So I want to just give a quick special thanks and again, introduce Dr. Peter Abadir, who is an associate professor in the Department of in the Division of Geriatrics in the Department of Medicine, and look forward to their introduction, and look forward to the exciting talks that will be given by a select number of colleagues of ours from both the School of Engineering and the Department of Medicine on their outstanding research achievements for the year. So I thank you all, and please, any other further instructions will include for the speakers today when we approach the question and answer sessions. Please remember to address your questions in the chat box. They will be moderated, they will be reviewed and asked in order of appearance. We will try to get through as many of those as possible, and encourage all of you to try to take as much time to participate in all of the events today, because I really think that you will find that this is going to be hopefully an outstanding research retreat this year. So with that, again, thank you all for being here today, and look forward to seeing all of you in the future. And so I will now turn it over to Dr. Abadir to begin his introduction and our transition to the Spotlight on Research.
Thank you, Nick, for the introduction. Good morning, everyone. My name is Peter Abadir. I'm an associate professor of medicine in the Johns Hopkins Geriatrics Division. On behalf of Dr. Anderson, Dean Schlesinger, and the Retreat Committee, we are delighted to bring you the Spotlight on Research, brief presentation of top-rated research. This fast-paced session will showcase some of the past year's best science that came out of two of the major entities at Hopkins, the Department of Medicine and the Whiting School of Engineering. This session was designed and executed in collaboration with Dr. Stephen Cohen. Dr. Cohen is an associate professor at the Johns Hopkins Carey Business School and a leading authority on persuasive speaking and effective presentation skills. He is well known for helping leaders communicate with authority, confidence, and influence.
Thank you, Peter. Good morning, everyone. Sir Francis Darwin once said that in science, the credit goes to the person who convinces the world, not to the person to whom the idea first occurs. Each of our speakers this morning has a formidable challenge: to convince us that the research matters in just 5 minutes. It's much easier, of course, if you have 3 hours to explain a paper. But how can you get the most important ideas across in only 4 slides? And how can you collapse 20 to 40 figures into a cohesive, accessible presentation? Well, our speakers this morning will do just that. The secret, as you will see, is to focus much more on outcomes, that is, the results and conclusions, and much less on process, the methods and analyses. Each speaker will share a bolt of insight, a finding or key idea that will make their research jump off the page. In doing so, these speakers will ensure that the best science is delivered in the most dynamic way, with power, with poise, and with passion. Now it gives me great pleasure to introduce our first speaker, Dr. Alexis Battle. Dr. Battle is an associate professor in the Department of Biomedical Engineering. The title of her talk is Integrative Data Analysis for Personalized Genomics. Please join me in welcoming Dr. Alexis Battle.
Good morning. So the premise of the work that I'm presenting is that every one of us has DNA sequence changes or variants that influence our health, but for which current methods are simply not sufficient to predict their effects. So I'll speak to both pieces of that statement. So first of all, the human genome varies. It varies from person to person. Each individual, in fact, harbors millions of genetic sequence differences that would distinguish them from their Zoom neighbor. Within those, each individual has around 50,000 genetic variants that are seen among very few other people in the population. That is, we call them rare. And within that, each person even has hundreds of genetic variants that we've never seen before. And this is referring to healthy individuals. So the second part of my statement was that genetics affects the health of every individual. So to give you some context, about 5% of Americans have a recognized rare genetic disorder. So these are the more extreme or severe cases of genetic disease, and this mostly affects children. But really, we all lie on a spectrum of genetic risk for disease. Many of these diseases are in fact influenced by thousands of genetic loci each. So really, none of us have zero genetic risk. But the bad news is that current methods, both experimental and computational, are simply insufficient for interpreting the full personal genome. So for around half of rare genetic disease patients, the sort of more severe cases that I mentioned, the responsible genetic variant cannot be identified, even though we suspect they have genetic disease. And for common diseases, we have only found at this point only a very small fraction of the genetic loci that we believe to be responsible for their observed heritability based on family studies. So overall, the situation currently is that we simply cannot predict or estimate the effects of most possible DNA sequence changes in the human genome. So I'm going to focus in this talk on two of the key reasons that personal genomics remains so challenging. And the first is simply due to the structure of the human genome. So only a tiny fraction, around 2% of human DNA sequence actually lies within a gene. And these are the more familiar units that encode for a protein that we can interpret pretty directly. The rest of the genome, about 98% of it, is what we call non-coding and is extremely hard to interpret currently, with no really straightforward code to refer to understand what it would do, but in fact can still have large consequences to our health. And the second reason it remains so challenging is that I mentioned these tens of thousands of rare variants that each person has. Well, these rare variants are not amenable to large association studies. So even as we collect large-scale genome sequencing data sets for many diseases, if you're the only person that I've seen that has a particular genetic sequence variant, I can't refer to these large correlational studies to estimate its effects. So the central idea inspiring our work to address some of these particular challenges is that any genetic variant that influences our health, regardless of whether it's rare or common, and regardless of whether it lies in these challenging non-coding regions of the genome, usually also causes observable molecular changes in the cell to proximal and predictable molecular readouts. So personal molecular profiling, along with personal genome sequencing, can offer direct evidence of these genetically driven perturbations, even for rare non-coding variants. And now there are readily available technologies that can read out quantities such as RNA levels, protein levels, and epigenetic changes that may result from these rare non-coding variants. So my lab has in particular taken this idea and developed novel machine learning methods that can integrate personal genome sequencing with personal molecular data to ultimately predict the impact of genetic variants from each individual's genome. So recently, we've specifically developed a method called Watershed that is an unsupervised probabilistic model that integrates multiple sources of information, including everything we know ahead of time about the human genome from population studies, in addition to the individual's whole genome sequence, along with personal whole transcriptomics or personal readouts of their RNA levels. This method is actually extensible to sort of arbitrary data types and ultimately can prioritize variants that are most likely to have a large effect on this individual, given any data that we've been able to collect for that specific person. So using Watershed, we've demonstrated that adding these additional diverse molecular data really dramatically improves the prediction of which DNA sequence changes are likely to have large effects, well over interpreting from genome sequence alone. We've actually experimentally validated some of these predictions using CRISPR-Cas9 editing to introduce these variants into cells in vitro and confirm their consequences. So what I really want you to take away from this is that all of us have genetic differences that could affect our health, but are currently extremely hard to assess. But using diverse molecular data along with machine learning methods, we can take steps to address some of the biggest challenges in personal genomics. And with that, I'll end and thank you.
Fascinating talk. Thank you, Dr. Battle, for a wonderful presentation. Next, it's my distinct pleasure to introduce Dr. Melanie Dispenza, Assistant Professor of Medicine in the Division of Clinical Immunology and Allergy and Clinical Immunology. And the title of her presentation today is Novel Strategies for the Prevention and Diagnosis of Anaphylaxis.
All right. Hi, everybody. Can you hear me? Perfect. All right. Thank you so much for having me today. Let's get right to this. As an allergist, I am most interested in IgE-mediated reactions, including anaphylaxis, which is a systemic allergic reaction to substances like foods or drugs. And unfortunately, anaphylaxis is still potentially fatal in modern times, despite prompt and thorough medical treatment. And there are no known therapies that can prevent anaphylaxis. We use steroids and antihistamines out of desperation, but we cannot prevent death from anaphylaxis, unfortunately. So that is what most of my work is focused on. If you can see here a schematic of a very broad overview of IgE-mediated reactions, mast cells and basophils have IgE on the cell surface, which when they see their specific protein, cause cell activation, inflammatory media release, and then those cause symptoms. My research focuses on interrupting this IgE pathway to prevent IgE-mediated reactions, including anaphylaxis. If you zoom in on this pathway, there's one particular enzyme that is the focus of a lot of my research called Bruton's tyrosine kinase, here with the red arrow, or BTK. This enzyme is essential for IgE signaling in human cells, and because it's also important for B cell signaling, it's been targeted pharmacologically for the treatment of B cell malignancies. And these now FDA-approved therapies are actually pretty well tolerated. So much of my research has been trying to leverage these therapies for a new indication of treating allergic disorders. And what we have found is that BTK inhibitors can indeed prevent IgE-mediated signaling in both mice and humans. This is data showing anaphylaxis in a humanized mouse model. And we use IgE plus then an IV dose of antigen to anaphylaxis mice. And as you can see in red, they have a core body temperature drop, an increase in clinical score from a scale of 0 being asymptomatic to 5 being dead. When you give these mice 2 doses of acalabrutinib, 2 clinically relevant oral doses, just hours before challenge, they have no clinical response whatsoever. And in fact, they look like the mice that are sham sensitized or the ones that don't get IgE. But perhaps more importantly, these 2 doses of acalabrutinib are significantly protective against death during severe anaphylaxis when we increase the dose of antigen, which is enormous for the field. We've taken this into humans. We did a small clinical trial where we gave food allergic subjects 2 doses of ibrutinib, and we did skin tests and basophil activation tests before and after ibrutinib therapy. As you can see, ibrutinib reduced every single skin test to the foods that they were allergic to, and 44% of all skin tests became negative. So we are currently extending this, trying to do clinical trials showing that BTK inhibitors can prevent clinical reactivity in humans when they eat the food that they're allergic to. And then I want to briefly mention a sort of side project that I'm working on that involves using microparticles as biomarkers for anaphylaxis. Another challenge with anaphylaxis is that it's a clinical diagnosis and it overlaps with a lot of other syndromes that we see. And no biomarkers are both sensitive and specific for it. And we use tryptase a lot, but it is notoriously not very sensitive, especially for food-induced reactions. Enter microparticles. These are tiny extracellular vesicles that are released by the plasma membrane when a cell is activated or injured. And because they express cell-specific molecules on their surface, you can detect them by flow cytometry. So we hypothesized that you might detect circulating mast cell and basophil-specific microparticles in the plasma after someone's had an allergic reaction or systemic reaction. And so we analyzed plasma samples taken from a walnut allergic man who had just ingested a dose of walnut flour and had a systemic reaction. And we found that we could indeed see peaks of mast cell and basophil specific microparticles in his circulation at different time points after this food exposure. So this is actually an N of 1, but we're very excited. So we just wanted to show it today. So in summary, if BTK inhibitors can prevent IgE-mediated systemic allergic reactions in humans, this would be paradigm shifting for the field of medicine. It's never been done before. And it could open up avenues for drug desensitizations, for food oral immunotherapy, making these both safer and more accessible to more patients. And finally, microparticles might be a novel biomarker for detecting food-induced reactions, including anaphylaxis. Thank you both so much.
Thank you, Dr. Dispenza. As a reminder, you're welcome to engage with our speakers this morning via the live chat and or ask any questions that you have. We'd love your participation. Next up is Dr. Jude Phillip. Dr. Phillip is an assistant professor of biomedical engineering, chemical, and biomolecular engineering, and is a core member in the Institute for Nanobiotechnology. Dr. Phillip's talk this morning, Human Aging at Cell Resolution. Let's welcome Dr. Phillip.
Good morning, everyone. Aging at cell resolution. Aging is inevitable. And while we gain 1 chronological year with the passing of each birthday, the biological outlook among individuals tend to be quite variable. An overarching question in my lab seeks to answer, why do we age differently? Why is it that among 3 85-year-old individuals, one can run a marathon One is bedridden and the other is in between. Questions like these become important, especially given the projected increases in our aging population over the next 30 years. It is projected that by the year 2050, 20% of the US population will be above the age of 65. And while this is great that people are in fact living longer, unfortunately, it doesn't always translate to persons living healthier. As a result, there are projected increases in socioeconomic burdens, partially stemming from increases in age-associated diseases, multimorbidities, and polypharmacy. But although these estimates highlight a significant challenges moving forward, as engineers, we would like to transform these challenges into opportunities. Opportunities to be creative, opportunities to be innovative, and opportunities to develop fresh new ideas. When I think about aging, I think through the lenses of scales, with each having a corresponding length and time component. This means that for every macroscopic feature, there is accompanied phenotypic changes that essentially spans from the molecular to the epidemiological, involving groups of genes and groups of people. This notion has led my lab to ask the question of which length scale potentially provides feasibility and promise to impact aging in humans. We propose that profiling biophysical properties of cells offer a potent solution. Here's why. As integrators of molecular signals, cells offer a mesoscale window into the aging process, with defective cells likely preluding the manifestation of age-related dysfunctions and diseases. Therefore, we believe that studying cells could allow us to map aging trajectories where deviations from healthy aging could inform us on age-associated diseases and disease states. In my lab, we're testing this notion. And in the process, we're developing new technologies, new frameworks to really quantify biophysical properties of cells. And by biophysical properties, I mean we're profiling how cells move, or what are their morphologies, or their capacity to generate forces. So far, we have found that yes, aging information is encoded in your cells. And we have shown the ability to predict the age of healthy individuals based on how cells move and the morphological patterns that they exhibit. In addition, we not only showed the ability to predict the age in healthy individuals, but when we included samples from individuals having age-associated diseases, these correlations broke. This approach also allowed us to stratify individuals into categories based on their aging patterns, to include patterns such as expected aging, which really means that they're aging in line with a larger aging population, plus or minus a standard deviation, or individuals who are aging at an accelerated rate, where their predicted age is larger than their chronological age, or delayed aging, where their predicted age is less than their chronological age. We're extremely excited about these results, and we are actively trying to push the envelope in our quest to impact people. As we build on this work, we envision a cell-based platform where we will not only be able to map and predict aging trajectories in favorable, in adverse, or in the in-between, but we envision a future where our technology will allow us to impact aging through the identification of strategies and approaches to modify aging trajectories for the better, In essence, we want to bring aging equity in living longer and healthier. Thank you.
Thank you, Dr. Philip. So the saying that age is just a number is not entirely true. Age is at least two numbers. Next, it's my distinct pleasure to introduce Dr. Elizabeth Luzak, Assistant Professor of Medicine in the Division of Cardiovascular Medicine. The title of the presentation today is How to Repair Failing Hearts.
Good morning. Thank you, Peter. Imagine you are being chased by your neighbor's dog, and your body activates the fight or flight response in order to enhance your ability to fight and protect yourself or to get away from the dog. Your heart activates signaling pathways to increase blood flow to the rest of the body to increase performance. Heart muscle cells use ATP as energy to increase heart rate and pump function. The main site of ATP production in the heart is the mitochondria, which have the ability to rapidly increase ATP production to support heart function. Now imagine that your heart muscle has been injured, perhaps by a heart attack that caused a portion of your heart muscle to die. In order to maintain normal function, the remaining heart muscle has to work harder and chronically activates some of the same pathways used in the fight or flight response. This chronic activation exhausts the heart's energy supply and ultimately leads to heart failure. Today, I'm going to tell you about how metabolic changes in the heart can drive heart failure. I'll introduce our lab's favorite signaling molecule, CAMK2, describe how it alters cardiac energy metabolism, and how heart function can be restored in a model of heart failure. The calcium calmodulin-dependent protein kinase 2, or CAMK2, is activated by increased intracellular calcium, which is required for muscle contraction and is an important second messenger in the fight or flight response. CAMK2 is known to be activated in models of heart failure, and we recently found that CAMK2 is a resident protein in mitochondria and that its activity is increased in mitochondria following cardiac injury. To test whether increased mitochondrial CAMK2 is sufficient to cause heart failure, we developed a genetic mouse model that overexpresses CAMK2 in heart mitochondria. These mice develop heart failure characterized by reduced function. Here in these MRI images from live animals, you can see that the normal heart contracts well to pump blood to the body, while the mito-CAMK2 transgenic heart does not contract well and cannot pump blood as efficiently. Next, we wanted to know what CAMK2 is doing in mitochondria. We used mass spectrometry techniques to identify potential targets of CAMK2 in heart mitochondria. We identified several components of energy production, including TCA cycle and oxidative phosphorylation. The TCA cycle primarily generates NADH that serves as an electron donor, driving the synthesis of ATP via oxidative phosphorylation. We found that CAMK2 in the mitochondria enhances NADH production by augmenting the TCA cycle activity, but it also impairs oxidative phosphorylation and ultimately leads to reduced ATP in these hearts. To determine if the reduced ATP was the cause for the heart failure we observed in the mitochondrial CAMK2 mice, we used two methods to replace ATP in the heart muscle. First, we directly added back ATP to isolated heart muscle cells by making a small hole in the cell membrane with a glass pipette. This restored the cell's ability to complete the ATP-dependent calcium cycling necessary for the muscle contraction and relaxation. Second, we used a genetic approach to enhance ATP levels in the heart by increasing the expression of mitochondrial creatine kinase, which functions to shuttle ATP out of mitochondria for use throughout the cell. We found that increasing mitochondrial creatine kinase expression restored the ATP levels in the mito-CAMK2 transgenic heart and also restored heart function, shown here in this MRI of interbred animals. In summary, we found that CAMK2 contributes to the energy deficiency associated with heart failure and that restoring energy to the heart improves heart function. Currently, we are developing new tools to identify targets of CAMK2 in mitochondria to help us understand the physiological role of CAMK2 activity in regulating mitochondrial metabolism. We hope these discoveries will aid in the development of treatment for heart failure patients to enhance cardiac energy production and restore or preserve their heart's function. Thank you for your attention.
Thank you, Dr. Luzak. That was fascinating talk. Next, we will have a 10-minute break before we continue with the rest of the speakers. I encourage you during this time to please go to the chat room, ask questions, engage with these speakers, and we will see you in 10 minutes. Thank you.
Welcome back, everybody. As a reminder, we hope you'll continue to engage with our speakers this morning. Feel free to pose questions in the chat box so that they can engage with you and you can engage with them. Well, let's continue with our excellent presentations this morning. Our next speaker, Dr. Sridevi Sarma. Dr. Sarma is an associate professor in the Department of Biomedical Engineering. She's also the Vice Dean for Graduate Education in the Whiting School of Engineering. Please welcome Dr. Sarma.
Thank you for giving me the opportunity today to talk about fragility in the brain. So when is half a brain better than the whole? It seems like an absurd question, but actually it's not so absurd because it's when the other half causes intractable seizures and when the remaining half can rewire to recover function, which is often the case in children. This is a hemispherectomy, and although it's rare, surgical resection is the gold standard to treat intractable epilepsy. 65 million people in the world have epilepsy, and 30% are drug resistant. They have intractable seizures. There is a potential cure, which is to successfully treat the epileptogenic zone, or the EZ, the source of the seizures. However, surgical success rates average 50%. And the main reason is because there's no real clinically validated biomarker of the EZ. We developed EasyTrack, an EEG analytics solution that computes a novel biomarker of the EZ called neural fragility, and when used to predict surgical outcomes, achieves an accuracy of 76%. So to appreciate the need for such a biomarker, let me walk you through the journey to diagnose and treat intractable epilepsy. So once a patient actually is deemed to be drug resistant, then the next option is, are they a surgical candidate? The idea here is the patient comes into an epilepsy center, stays there for at least a week to be monitored, and a plethora of neuroimaging data is captured from that patient's brain, including scalp EEG. The idea here is to try to narrow down where is this epileptogenic zone, and is it in a focal area in such a way that I can surgically treat it. Often that's not precise enough, so the patient undergoes a next phase of monitoring, which is invasive. Here they get electrodes surgically implanted into the brain to capture intracranial EEG data. They recover from the surgery and stay in the hospital for up to 2 weeks. Why so long? Because a clinical team is waiting for that patient to have several seizures so that they can go back and visually inspect the intracranial EEG data channel by channel to see which areas of the brain, which channels are exhibiting some abnormal activity right before the onset of a seizure. Those channels might pinpoint to the true EZ. A surgical plan is made, surgery is done, and here we are today still at an average of 50% success rate. Our solution, EasyTrack, computes a new biomarker called fragility. And we're going to talk about what fragility is. But let me just tell you about the solution itself. It takes in a snapshot of EEG data, builds network models from the patient's EEG recordings. So these are patient-specific dynamical network models, and produces a really simple, easy-to-read heat map. We call it a fragility map. And the idea here is on the y-axis, these are regions of the brain channels, and this is over time. And what you can probably see and what you're intuitively thinking of is right before that seizure event, there are certain channels that become red hot, very fragile, right before the seizure starts. These are the areas that are highly likely to belong to the epileptogenic zone. So what is fragility? I'm going to give you an intuitive definition. So when you have a healthy brain, there is a notion of balance between excitatory neurons and inhibitory neurons. There's a balance. But when you have epilepsy, one or more nodes in the network, in the brain network, cause an imbalance. Why? Because their functional connectivity, their influence on the rest of the brain changes in such a way that seizures can be now triggered in the brain. Fragility of the node in the brain is the amount of this balance. It's a quantification that we can compute from a dynamical network model estimated from that patient's EEG. Here are some examples. On the left is a fragility map overlaid on the patient's brain. What you can see in the bottom is the surgically treated area outlined in black. And what you can see in this case, the fragile areas were surgically removed, and this patient had a successful outcome. On the other case, you see no overlap between the surgically treated region on the left and the fragile areas that are more on the right. This was a failed outcome. When you use this measurement of overlap between the fragility maps and what the clinicians treated, you can use that overlapping measure to predict surgical outcome. So to validate fragility, we applied fragility maps on 91 patients that were treated across 5 different epilepsy centers, and we were able to achieve a prediction accuracy of 76%. And in this population of 91, there was a 48% success rate. We have since, this retrospective study is currently being reviewed in Nature Neuroscience. Because it has a high clinical impact or potential for high clinical impact, we started a company to commercialize EasyTrack, and it was recently FDA cleared in December 2020. Thank you.
Thank you, Dr. Sarma. It's just simply refreshing and amazing how when engineers and clinicians work hand in hand, just awesomeness happen. I have tons of questions, and I want to encourage everyone that has questions to go to the chat room and post the questions, and we will have possibility of answering live the questions. Our next speaker, I am pleased to introduce Dr. Florin Celaro, Associate Professor of Medicine and the Director of the Meyerhoff Inflammatory Bowel Disease Center, and the title of the talk today is Shape-Changing Microdevices for Extended Drug Release.
Morning, and thank you for the invitation. It's very hard to follow Dr. Sarma's presentation, and if I were to fail too miserably, then I'll be looking for a postdoc in her lab. So, anyways, I'm Florin Celaro. I'm in the division of gastroenterology, and the title of my talk today is Shape-Changing Microdevices for Extended Drug Release. So as you all know, extended release drug formulations improve compliance with medications and patient outcomes. So the question becomes why there are so few orally available extended release drugs. And part of the explanation is because the GI tract is actually made for food intake. It's not made for drug delivery. So there are a couple of features that I would like to point to here, which are, first, if we were to have a chemical platform that is able to release drugs over, say, a period of a week, and if we put it on this conveyor belt, it would actually not have the chance to release the drug over said week because it would be pushed out with the rest of the food intake. The second feature here is that if you have too bulky of a device, it might actually get stuck and the whole conveyor belt stops, and our colleagues in surgery tend to dislike that quite a bit. So basically, we aimed to design and manufacture micro devices that are able to somehow latch onto the inside of the gut, that are small, so they don't cause an obstruction. They need to be able to unlatch And we also wanted no wires and no batteries for packaging reasons and others. The inspiration, one facet of the inspiration came from hookworms. And if you have not seen this ugly beast to the right here before, this is what a hookworm looks like. And it has the ability to latch onto the intestinal mucosa, and that's part of its life cycle. Another side of our inspiration came from the human hand, and this is a human hand in an open position, and of course, upon closing, the hand can actually latch onto things. And this is the device that we created. As you can see, it has a central portion just like the human hand. This is actually where we deposit the drug that will be released slowly. And these are the fingers that actually connect to the intestinal mucosa. This is the result of about 12 years of collaboration with Professor Garcias in the School of Engineering. And we recently were featured on the cover of Science Advances. And the reason I'm showing this is also because it demonstrates our thought process. We figured that if we make these small, tiny devices that are able to autonomously bind to the intestinal mucosa and sit there for a while, then they will be able to allow the chemical part of them to release the drug in an extended fashion. So here to the left, we have the design of the grippers. There are a couple of important features without getting too much into details. So these devices are metallic and they have intrinsic stress, and they want to actually curve and close towards the weaker side, which is here, the superior side. To keep them open, we add the thermoresponsive trigger, which is here in purple. And the thermoresponsive trigger that's deposited on the weak side keeps it open. However, it responds to biologic cues, and when it does so, the gripper closes. This is an image of a gripper that shows the central portion where we deposit the drug, and these are the fingers that upon actuation, they will close upon the mucosa. This is such a device that is in a closed position. And just to give you a sense of the size, this is a 27 gauge needle for venipuncture, and this is the size of the devices. This is how they're made on wafers. And I wanted to show you a video of how they close. They are here in suspension, and we add to the solution a biologic cue that will degrade the layer that actuates them. And if you want to watch them, we will see them closing at about the same time. There is this one guy here who refuses to close one of the fingers, but outside of that, pretty much all the devices closed as they were designed. And this other video here shows such a device that is closed and it's latched, and you see the fluorescent small molecule that is getting released slowly from the device, which is exactly what we wanted to see. In a proof of principle study, we wanted to see if we can release ketorolac in an extended release fashion in vivo. So for that reason, we gave some of these devices by a rectal route. I'll get back to that in a second. And you see an X-ray here that shows that these devices are actually closed onto the mucosa in this animal model. And this is an important graph that shows that the pristine small molecule tends to have a concentration that falls off rapidly when it's given as a pristine drug. However, when it's given with these devices, we are able to actually extend its release quite nicely. And these are the numbers. So we increase the area under the curve, which is equivalent to the exposure of the drug, by twofold, and we increase the half-life by about sixfold. So in conclusion, what I have shown you today is that we design shape-changing micro devices that have no batteries, no wires, that respond autonomously to biologic cues. And we can encode those biologic cues based on where we want them to close and when. And we have shown in a proof of principle study that we can extend drug release in vivo. Of course, we are hard at work now towards developing a capsule to protect these devices so that we can deliver them by mouth. The team has many members, but I would like to express my special thanks to Professor Grosias and to Arijit Ghosh, who started with us as a postdoctoral fellow and he's now on faculty, to Lin Li, who's in my lab and who started as a student, moved on to a postdoctoral position, and now she's on faculty, and of course, Rana Rice in the Johns Hopkins Drug Discovery Center. With that, thank you, and I would love to see some questions in the chat window.
Well, thank you so much. Really interesting presentation. Our next speaker is Dr. Suchi Saria. Dr. Saria is the John C. Malone Associate Professor of Computer Science, Statistics, Nursing, and Health Policy. Dr. Saria is also the Director of the Machine Learning, AI, and Healthcare Lab, the Research Director of the Malone Center for Engineering and Healthcare, and the founder of Bayesian Health. Dr. Saria's presentation this morning is Safer in Proactive Care Enabled by AI ML. Let's welcome Dr. Saria.
Thank you so much. While I'm presenting today, this is really a body of work with contributions from at least 50 wonderful, diverse collaborators. Today's care is very reactive, and it doesn't need to be this way. So there are complications like sepsis, pressure injury, readmissions that we could have potentially prevented if only we could have anticipated it. And these events also lead not only to poor patient outcomes, but a lot of financial waste. My research focuses on how do we use data and advanced algorithms to enable proactive care. So let's walk through an example. Back in 2015, we at Hopkins did this piece of work where we first showed it was possible to identify sepsis, which is one of the leading causes of in-hospital mortality, identify sepsis early by combining labs and physiology that are available in your EMR. This paper spurred a lot of interest in the field with upwards of 200 papers from various institutions. We've simultaneously also been very hard at work since. So the initial result we published was on the ICU cohort. Since then, we've expanded significantly from ICU to inpatients and ED floor and ED patients, going from just model development research to how do we deploy these models in practice? How do we get adoption and engagement? And then as we developed this, we had to license this research out. So we had a 20-month data embargo that was on all outcome study that was recently released. So I'm actually pretty excited to show you guys some outcome data. So what have we learned? So the first thing is, first, on a very large data set of 270,000 encounters from 4 sites over 4 years, the model is actually quite accurate and precise. So let me give some context. Sepsis is a needle in a haystack problem. Getting anything about with high sensitivity is upwards of 0.7 is very hard. And then getting above 0.1 ppb is very hard. So we've been able to achieve very high sensitivity and ppb in both cases. 2, in sepsis, every hour counts. So what is our lead time? Turns out in 63% of cases, the alert fires before physicians would have gone in and prescribed an order. In 41% of cases, there's more than 1 hour lead time. In 13% of cases, there's more than 12-hour lead time. So what does that mean in practice as we deploy the system? So it turns out, in practice, we see similar performance. So we do see 61% of alerts translate into earlier detection and prior to physician suspicion. How do the physicians behave in response to it? Turns out 46% of patients do receive care from physicians in response to the alert, within 1 hour, they're prescribed timely treatment. In 72% of cases, they get it within 3 hours. And how does this all now turn into outcomes? So this is perhaps the most exciting and personal result, which is when we analyze data looking at patients who receive timely care versus delayed care. So those within 3 hours versus post 3 hours, risk adjusted, we saw 5% reduction in absolute mortality for sepsis. And this translates to 27% reduction in mortality rate. Here, I've also listed confidence intervals to say this is significant. How does this compare to everything else that's out there? So there's a lot of data here, so highlights. First, in terms of sensitivity, positive predictive value, which is very, very crucial for gaining physician trust and adoption, TRUES is able to achieve upwards of 0.5 sensitivity, which is very hard to get, and significantly higher PPV than any other system out there. And in terms of outcomes, there are a couple of other outcome studies. Because of the lead time we have and the accuracy, which then translates into adoption, we've been able to get significant outcomes. Let me contrast this with one system here, Dascana, where they were able to get good outcomes, but they had to put dedicated bedside resource, dedicated centralized resources to monitor the system because the amount of noise that the system produced. So why are we able to achieve all this? In a nutshell, in order to do machine learning well and accurately, there are three components that matter: the quality of the inputs, the quality of the phenotyping, and the quality of the learners. And we've published extensively in these three areas, and by combining strategies, you can actually improve precision by 300 to 700%, like we saw in the case of sepsis. My final slide. So in 2018, like I mentioned, I helped found Bayesian. We licensed out this work to a spinoff out of Hopkins, have built a very talented team and applied this work across now to a number of other areas and have been able to achieve state-of-the-art results in multiple clinical areas. So I think there's very significant outcome for improving, there's very significant opportunity for improving outcomes by activating proactive care, and high quality machine learning solutions will be a crucial component of that.
Thank you, Dr. Saria. This is fascinating, and it reminds me that I quote Dean Schlesinger in that engineering next steps is healthcare, artificial intelligence, machine learning. I think we will see very different medicine in the next few decades. Next, it's my distinct pleasure to introduce my colleague, Dr. Janet Salicano, Associate Professor of Medicine and Infectious Disease. Dr. Salicano's talk today will be on mechanisms of HIV-1 persistence.
Thank you very much, Peter. Amidst the coronavirus pandemic, we forget that there's 38 million people infected with HIV, and most will die from AIDS without lifelong antiretroviral therapy. 25 million people are now on therapy, and only 2 people have been cured. Antiretroviral therapy produces a rapid exponential decay in viremia to below the limit of detection and prevents disease progression. But no matter how long people remain on antiretroviral therapy, viremia rebounds when antiretroviral therapy is interrupted. And this rebound is due to a small pool of infected cells that have a very long half-life. And these are lately infected resting CD4 T cells, and they were first described at Hopkins. And these cells arise as a consequence of normal T cell physiology. HIV replicates in activated CD4 T cells, and these cells die very quickly. On rare occasion, however, HIV will infect an activated CD4 cell as it's transitioning back to a resting state, a state that's non-permissive for viral gene expression. And this results in a stably integrated but transcriptionally silent form of the viral genome in a long-lived memory cell, a perfect mechanism for viral persistence. And now if that lately infected cell gets activated in the future, it can produce virus. Now, lately infected cells can be detected with a viral outgrowth assay in which latency is reversed by T cell activation. Although these cells are rare, about 1 in a million with the viral outgrowth assay, we showed that this pool of lately infected cells decays very slowly with a half-life measured in years, not days. And so therefore, this latent reservoir provides a long-term reservoir for HIV and is the major barrier to curing HIV. Now, we've analyzed the landscape of proviruses that persist in treated patients and the reasons that the reservoir is so stable. The landscape of persistent proviruses turns out to be dominated by defective proviruses, including those with large deletions, shown here in white. Cells with defective proviruses constitute a large fraction of the total population of infected cells detected with standard PCR assays and greatly outnumber cells with replication-competent proviruses that we detect with the viral outgrowth assay. However, we've also discovered using whole genome sequencing that there is a significant number of cells with genetically intact proviruses. We've shown this using whole genome sequencing. And these genetically intact proviruses are not readily detected in the viral outgrowth assay, but could contribute to viral rebound when patients stop antiretroviral therapy. So therefore, these genetically intact proviruses in the blue circle represent the real barrier to cure. And we've recently developed a precise and scalable assay to measure them. The intact proviral DNA assay separately quantitates proviruses with defects in the 5' and the 3' end of the viral genome and the intact proviruses that preclude cure. We think this will be the first FDA-approved reservoir assay and will be essential for evaluating interventions to reduce the reservoir. Now, most recently, we've been focusing on two concepts related to reservoir dynamics and viral rebound. And what we've shown is that among cells with replication-competent proviruses, there are clonal populations of infected cells that wax and wane over time. And this proliferation of infected cells is driven mostly by antigens. For example, among infected CD4 T cells that are specific for CMV, we can find very large clones. And each cell within that particular clone has the identical provirus integrated into exactly the same position within the human genome. This result can only be explained by antigen-driven proliferation, which generates large clones that dominate the latent reservoir. This proliferative process is the principal reason that the reservoir does not decay, but it will be difficult to target because it's a fundamental aspect of normal immune responses. Interestingly, several studies have shown that the clones detected in the reservoir with viral outgrowth assays are rarely observed among rebound viruses seen on treatment interruption. We've recently demonstrated that this reflects the fact that outgrowth of many of the viruses in the reservoir is actually blocked by autologous antibodies. For example, among the diverse viral variants present in the reservoir in this infected individual, only the variants in orange are capable of growing out in the presence of autologous antibodies. In contrast, this large clone is extremely sensitive to neutralization with autologous antibodies, and thus the fraction of viruses in the reservoir that actually contributes to viral rebound is smaller than originally thought, and we just need to prevent growth of viral variants that are not readily controlled by host immune responses to have a functional cure. And so to conclude, a latent reservoir prevents HIV cure. Accurate reservoir measurements require distinguishing intact from defective proviruses. Reservoir cells can proliferate in response to antigen, and some reservoir viruses are neutralized by autologous antibodies. My husband Bob and I have been working together on these problems for many years, and I'd also like to acknowledge the very talented graduate students in our lab who contributed to the studies that I presented today, including Greg Laird, Joe Varial, Lynn Bertinoli, and Francesco Simonetti. Thank you.
Thank you so much. Thank you so much. Great job. Let's give another round of applause, or at least a virtual round of applause, to our 8 outstanding speakers this morning. Truly impressive. Thank you so much. Well, we're a little bit ahead of schedule, which is great. So we have time for a few live questions before another short break. Our first question is for Dr. Selleru. One of our audience members says, neat. How do these devices get out of the body and after how long? And are they MRI safe?
Thank you, Dr. Brodman. That's an excellent question. They are not MRI safe at this point, but we have data that says that 100% of them get out of, we've done some experiments in swine, they are out of the body in a week, and the reason for that and the way they unlatch is based on the fact that the intestinal mucosa renews, and we can fine tune the depth of the fingers So basically, we can make them come off sooner or later, depending on how deep they grip the mucosa. We can also build in another layer of biologic cue that would actually be able to open them to assist with unlatching. Thank you for the question.
My next question is for Dr. Sarma. And the question is, what you presented is amazing and opens the questions for, can you translate the same technology and tool to other organs? Fragility is very similar to the word frailty. Can you see the use for that for muscles, EMG, atrial fibrillation?
So the concept of fragility is thinking of a weak link in a network. So when you think of any biological systems where you have many elements, whatever those are, that are functionally influencing each other, okay, then to form a legitimate network, then there is a notion of fragility, which areas of which nodes in that network are fragile. But fragile in the seizure application is a stability phenomena. So there's actually theory behind this that says, I have this brain, it's a dynamical system, and when you have a seizure, it goes unstable. So a fragile node is something that will more likely cause instability. So in other contexts, you have to ask the question, what does it mean to be fragile? What is the phenomena that's causing the disease or the symptom? And then translate that back into what does that mean in terms of the property of a dynamical network? Does that make sense?
Yes, thank you.
Great. Well, thank you so much. We're out of time. It's now 10:50 on the dot. So we're going to take a 5-minute break and then come back and continue our program this morning. Thanks again to my wonderful colleague, Dr. Peter Abadir, and all of our speakers and everyone who made this virtual conference possible. We'll see you at 10:55 a.m. Thanks so much.
It's a pleasure to start us back at the second part of the first day of the Engineering Department of Medicine Research Retreat, and it's a particular honor and thrill to be able to introduce the next speaker. Dr. Tekjeep Ha is a person who uses innovative, really remarkable physical methods to measure and visualize single molecule behaviors with fundamental importance to biological processes. By listening, you'll hear this. I'm just going to touch on some of his very storied career. But Dr. Ha grew up in Korea. He attended the Seoul National University where he studied physics. He then obtained his PhD in Berkeley, a process that was interrupted by a year of national service in Korea. After his PhD, he did a postdoc at the Lawrence Livermore National Laboratory, and then became a faculty member at the great engineering school at Champaign-Urbana, where he was in biophysics, physics, and computational biology. He is a Bloomberg Distinguished Professor, so one of a really rarefied cohort of people at Hopkins. He's an HHMI investigator and is a member of the National Academy of Science. He served as an editor for Cell and currently serves as an editor for Science, and he's won a whole list of awards, and I'll just mention a couple of them. The Michael and Kate Barani Award at the Biophysical Society, and the Hoam Prize for Korea. Some have described this as the Nobel Prize of Korea. I think, on a more personal level, TJ is somebody who brings a lot of excitement and fun, passion to science. And I think he represents, as a person, a lot of the potential not fully realized yet at Johns Hopkins, where diverse kinds of interests in physical science, in basic biological science, and clinical science can come together to create really exciting, innovative things. I think working at the interfaces is what he does so well, creating measurement tools that were never imaginable before. And I also want to thank him for spending time interviewing and meeting with our residents who are truly interested in a scientific career. So it's my great pleasure to welcome Dr. Ha.
Thank you very much for the kind introduction. And also like to thank the organizers for inviting me to speak at this wonderful meeting. So I'm a biophysicist by training, and I moved my lab to Johns Hopkins in 2015 with the hope of bringing advanced biophysical technologies closer to medicine. We are making steady progress, and I'm hoping to make additional connections with scientists in medicine and also engineering in the future. So when I was first invited by Nick, oh yeah, this is a meeting some years ago that Stephen Chu came to give a keynote speech, and then I realized that that was just last year. So it feels like a decade ago. So in fact, I came to meet him at his appearance with Stephen Chu. That was almost exactly a year ago. And in retrospect, it was a very an innocent time. And hopefully, we are seeing the end light at the end of the tunnel, and we'll be able to get together in the next retreat in person. So today I will talk about CRISPR. You can think of it more as an opening gig for our keynote speaker, Jennifer Doudna. CRISPR stands for Clustered Regularly Interspace Short Palindromic Repeats. That's quite a mouthful. And the most famous enzyme is Cas9. And it is a new technology for genome editing, but perhaps not anymore because, of course, we all celebrated the Nobel Prize in Chemistry last year, shared by Dr. Doudna and Dr. Charpentier. And so perhaps we shouldn't call it new anymore. Maybe you can still call it young technological platform, because I think we still haven't seen half of what it can do. And there's a lot of room for additional innovations and improvement. So to quote Michelle O'Malley, who said that nature is the best engineer, but we are looking to speed up her process. I think this really applies to CRISPR. In fact, if nature puts her mind onto it, nature will always do better than we can. But some of the enzymes evolved without selective pressure to discriminate against billions of base pairs of DNA, because they evolved in bacteria with 1,000 times smaller genome size. This is the reason why, for example, we have off-target effects in CRISPR-Cas9, and engineers actually working very hard to improve on nature to make genome editing more specific, as an example. And the topic of my talk is more about speeding up the process of gene editing. Just to give you a bit of a background, the Cas9 protein is powerful for genome editing. And you can think of it as molecular scissors that can cut the DNA in the genome, but it can be programmed by using a piece of RNA called guide RNA that you can design the sequence so that you can base pair with the target DNA strand. And ideally, only that DNA will be recognized and cut by the nuclease domains making two cuts on the DNA. But once you make the cut, the cell will actually use the existing DNA repair processes to repair the break. And sometimes you repair that break in an erroneous manner, causing gene disruption. And this is a method that is actually widely used for screening experiments. But we can also provide a template DNA that can be used to repair the damage, to edit the genome precisely, we can also insert tags or fluorescence proteins at that location through this pathway. As we are pretty well aware, the technology may not be entirely safe. So there are two classes of safety issues. One is the off-target effects, where you cut the DNA not targeted by your guide RNA due to cleavage promiscuity. But even when you cut the correct target sequence, you can have undesired on-target effects. It can cause DNA damage response, activating p53. So that cells that survive the process actually have somehow inactivated cancer tumor suppressor gene, so that you could, in principle, actually cause tumor. You can also induce large deletions and complex rearrangements at the cut site that people only began to discover fairly recently. Now, another limitation of CRISPR technology is that the fact that the seizures that we are talking about here is asynchronous. We don't actually know or control exactly when cleavage occurs. And if you have a very broad time window when the activity can occur, then it's really difficult to study what happens afterwards in terms of DNA repair and in terms of DNA genome editing mechanisms and kinetics. So people in the lab actually set a goal to control CRISPR activity in micrometer length scale in seconds time scale. So Yang Liu is a postdoc who is supervised jointly with my colleague Binu, who is an expert in live imaging, and Raja is an MD PhD student who teamed up with Yang to really drive this project beyond my wildest imagination. We already know that CRISPR has extremely high precision along the genomic coordinate. Now they want to add precision in time and space. So we're going to call this technology very fast CRISPR because we think that a second's time scale is not fast enough to be called ultra fast. We allow Cas9 to bind the DNA target beforehand, but not cut the DNA due to a photocleavable blockade. And then you shine light to remove the blockade. And then, only then, the seizures will close and cut the DNA, and ideally very rapidly at that. So that is the concept. And this relies on two interesting properties of CRISPR-Cas9 shown here. So just to get into a bit the mechanistic details of the enzyme, the Cas9 protein shown in the back as a gray molecule binds to the guide RNA, and the rest strand denotes the sequence that targets the genomic DNA. And then it'll bind to a sequence called PAM in the DNA, and then use a binding energy to melt the seed region, PAM proximal region, just a few base pairs, and then compare that to the guide RNA by forming this RNA-DNA heteroduplex. If the matching is good enough, you can continue to unwind the DNA duplex and base pair with the guide RNA. And if the matching is good enough, going beyond 17 base pairs or so, then you can actually trigger a conformational change to induce DNA cleavage. And then once you make two cuts, creating a double strand break, then the cell will do the rest using the existing DNA repair processes. However, we also know that, again, based on mechanistic studies performed by many labs, including our own, that you don't need 17 base pairs for stable binding of Cas9 to the DNA. You only need about 10 base pairs matching in the PAM proximal region of the DNA. So we thought we could put a photocleavable caging groups in the palm distal side of the guide RNA, so that you allow Cas9 to bind stably to the target DNA, but not cleave because of the blockade. And then we shine light to remove the caging groups. Then finish the process of zipping between the guide RNA and target strand, and activating cleavage very rapidly. So that is the concept of very first CRISPR. And if you want, you have your Cas9 proteins pre-bound to the target DNA, waiting for your signal that you deliver using light. And then we can have synchronous cleavage and then easily start to study what happens afterwards. It works really well inside a living cell. Here I'm plotting as a function of time, DSP percentage, basically what fraction of DNA has been cleaved by that time point after shining light for some time, for like 30 seconds. In fact, even at the earliest time point of 30 seconds, we see that half the DNA has been cleaved in our assay. This is much, much faster by orders of magnitude than the previous record shown here, performed using chemically inducible Cas9, which took about an hour to reach the maximum effect, which is still lower than our activity. So this is great news. We can actually use the very first CRISPR to induce DNA cleavage at a precisely defined moment in time. There are many actually ways to generate genomic DNA breaks, including chemical mutagens, physical radiations, X-ray, and UV. You can have some endonucleases that are sequence specific, but compared to the technologies that are already available, our method creates a very well-defined break, chemically clean break, and we are very specific. But we can also target any sequence we want in the genome because we are using CRISPR-Cas9, and we are very fast, and we have also high efficiency. So we believe that the technology has a great potential in applications of studying DNA repair in cells and also in tissues, perhaps even patient tissues. So let me give you some examples of how we can use it to study DNA repair. DNA repair is actually a very complex process involving many, many different proteins. But there are actually several widely used markers of DNA damage response. So I'm going to be showing some data on 3 different markers. MRE11 that arrives very early to sense the DNA damage. And then there's another protein that arrives a little late. And then this is a histone modification that is widely used as a marker of DNA damage response called gamma H2AX. So what we chose to do at the beginning was to develop a protocol to perform ChIP-seq in a time-reserved manner. Let me explain what that is. So you shine light at t equals 0, and then you harvest the cells at different time points. And then you perform chromatin immunoprecipitation. So you use antibodies against your protein of modification of interest to collect the chromatin fragments that contain those interesting features. Then you do sequencing and map it to the genome to map where these changes occur as a function of time. And here's an example of performing time-reserved ChIP-seq of the early responding protein, MRE11. And you can see that when you make a cut in the middle, we're looking at 2000 base pair region of the genome, no light, no signal. At 2 minutes after light, you see a signal appearing, and this grows over time. You can quantify this and show that, surprisingly, Cas9 generated break can be detected by the DNA repair protein within 10 minutes, which is actually contrary to what people had expected. In addition, we can go into a bit more details. If you read the sequencing reads from Illumina sequencing machine that actually span that cut site, meaning that DNA has been already repaired and ligated, then you actually don't see anything like that in the first 5 minutes, but by 15 minutes, you already see the spanning reads showing that Cas9 generated breaks can be repaired really fast, within just about 10 minutes by the cellular machineries. There's another example of measuring changes in the chromatin, also as a function of time during the DNA repair process. This is a well-known marker of DNA damage response. And you make a cut, and then even at 2 minutes, you see a signal showing up, which grows in time and in width, in height. And you can also plot the width as a function of time, shows that it grows linearly in time, suggesting a one-dimensional mechanism of spreading of this modification. We can determine the rate of spreading is really fast, 150,000 base pairs per minute. And it's worthwhile emphasizing that we had no information about the spreading kinetics before because there was no way to synchronize the reaction previously. Let me also give you a few examples of using the technology to study DNA damage response in real time from live cells. So what we are doing here is actually to use late responding protein called 53BP1, but fused to M-cherry, that's a red fluorescent protein, to see that they are ganging up on the DNA damage sites in the same cell, that there are two clouds of red proteins showing up here. But in order to distinguish existing DNA damage that happened just spontaneously from the damage we create, we need to mark the site of DNA breaks. So using GFP fused to the Cas9 enzyme, and that's how we actually visualize it. But it turns out a single GFP is not easy to visualize. We actually have to use repetitive sequence nearby and using inactive truncated guide RNA to allow binding, but no cleavage and next door. But you have dozens of GFP lighting up the genetic locations clearly. And then you can see that these two sites that are cleaved also recruit DNA damage proteins. And you can also see that in the live imaging. So there are two green spots dancing around over time, and they separately recruit the red cloud because they are undergoing DNA repair process. You can take snapshots and show that this DNA site is being repaired, but once the repair is done, now the activated Cas9 can bind and then make another cut. You go through another round of repair and so on. And you can use these gray lines to denote different cycles of repair. And we have done this from many, many cells. And here's a tabulation of 60 single cell experiments and showing that the initial repair time shown in the gray lines is highly variable between cells. And many of the cells also show second and third round of repairs, showing additional cutting and repair going on, likely because initial repair was done accurately. Interestingly, we found that the first round of repair, this iteration, is much longer than the subsequent rounds. We think that something is happening to the chromatin to facilitate DNA repair so that damage occurring soon after can still be done much more rapidly than the initial round of repair. We can also look at the other alleles. You have two alleles in the same cell and see if they are any correlated. And in fact, there is a nice correlation suggesting that somehow large variations we see is due to an extrinsic factor like such as cell cycle differences and differences in DNA repair protein concentrations. So this is the kind of thing we can do now using the new technology. So I have told you that we now have a control over CRISPR activity in time, so we can make the cut when we want. If we can also control the activity in space where we want, we can then begin to claim that we can do genome editing in 5D, 1D for genomic coordinate, 1D for time, and 3D for space. So let me just illustrate our spatial control. By activating CRISPR at the single allele resolution. So you have two green spots marking two copies of the same gene. We can actually use the imaging of the green spots to guide our uncaging laser to that spot only. And ideally, only that spot will generate a DNA break and then recruit red cloud as the marker of the DNA repair process, not the other. And then you can claim that you have achieved a single allele resolution down to about a micrometer length scale. So we can do this on a repetitive locus. And among the two green spots, only this one recruits the red cloud because that's the only one that saw the light. And we can also do this on a single clean cut locus next to repetitive sequence. Again, only this one on the right side recruits the DNA repair proteins because that's the only one that saw the light. And you can do this many, many times, 100 times each, and show that in the vast majority of cases, only the site that saw the light stimulated recruits the DNA repair protein as not the other, showing that indeed now we have demonstrated CRISPR control at the subcellular and micron length resolution in space, and we can also control activity at the single allele level. So there are many really exciting applications we want to pursue. For example, we can do this on other parts of the genome that are interesting, like telomeres and ribosomal DNA. And these are some early data that students have acquired. We think that we can use the technology to perform single allele editing. And furthermore, because we are activating only a small fraction of the Cas9s inside the nucleus using imaging-guided activation, we think that we can eliminate altogether the off-target effects. And this is something that we are planning to test formally. Let me end by showing you a couple of related experiments that students have performed that got published last week in 2 papers. So if the first part of the talk was about using light to activate CRISPR, here we are trying to deactivate CRISPR using light. And there are many reasons for doing that, because if you have sustained cleavage going on all the time, even after gene editing has been done, those activities could also give rise to additional side effects. So you want to turn them off nicely and very cleanly. And we achieve this by using a photocleavable group in the guide RNA near this is 14th position. And so without light, you get regular Cas9 activities, but you shine light to cleave off this part, and then Cas9 becomes inactive. So we have a single component system with a light-controlled kill switch, and it works really well. So in terms of how well you can do inactivation based on the standard readout, basically we are eliminating activity better than 99% with a single dose of light. So 99% efficacy with a single dose. So I think that's better than Moderna, or that's a joke. And this is actually much better than competing technologies for inactivating Cas9 using anti-CRISPR protein small molecule inhibitor and another technology to use engineered RNA molecules. And in fact, we can then begin to use it to study DNA repair. And in this case, how quickly DNA recovers from the DNA damage response. So you shine light, and then our DNA repair protein then disappears once you can no longer make cuts inside the cell. So we think that's a really new approach that can be very exciting. The last bit of data I want to share with you is about a new genome imaging technology that we titled Goldfish. So DNA fish stands for fluorescence in situ hybridization. It's actually a standard technique for studying genome organization in cells, but also in making diagnostics in patient tissues. So what you normally do is to globally denature the genome by heating the sample to 80 degrees Celsius. But this could compromise ultrastructures inside a cell, but also because you are denaturing 3 billion base pairs of DNA, your fluorescent labeled FISH probes can bind to many of those sites, that are not correct sites. So you have a lot of background that can compromise imaging resolution and sensitivity. In contrast, goldfish, the acronym here is genome oligopaint via local denaturing fish. We denature the genome locally using a super helicase that we engineered. Helicase is an enzyme that unwinds the DNA into single strands. And we load that at a single Cas9 cut. And here's the illustration. So Cas9, we have a Cas9 variant that makes only one cut at the site of your interest. And then our helicase enzyme, called RepX, is loaded at that nick to unwind the downstream DNA. And then you generate single-strand DNA in the genome. Then you can then bring your FISH probes with fluorescent labels to visualize them inside the cell. And we show that this unwinding occurs on the length scale of about 2,000 base pairs. So instead of unwinding, melting 3 billion base pairs, we only melt 2,000 base pairs. So you have a lot fewer non-specific binding sites for your FISH probe. So we have very much reduced background, and that should really improve the resolution and signal-to-noise ratio. But also because we don't denature the genome by heating, we can ideally preserve the structures better. So that is the concept. And here's some of the images where we can show that goldfish can be used to see two copies of the same gene. In all of these cells, they show basically just two spots. We get extremely high signal to background ratio, about 10:1. And because our specificity comes from cleavage, which is a lot more stringent than binding specificity, as I illustrated earlier, we can also compare our technology to other imaging technologies that rely on Cas9 binding, shown here. So our method actually has a much better signal to background ratio by about an order of magnitude or better. This is a log scale here. And you can also see the difference just from the images. So our goldfish, they really barely see any background, but if you use Cas9 binding based assays, you see a lot higher background. So Yambo is the one who did the work. And then Yambo teamed up with another graduate student, Taylor. They acquired breast cancer tissue, never frozen tissue, through a commercial source and performed goldfish here. And the idea was to visualize HER2 gene amplification in HER2 positive breast cancer. Indeed, in sections of the tumor, you see HER2 signal is greatly amplified, whereas in other parts of the tumor, HER2 is not amplified, and you see only 2 spots of HER2 per cell. So, and we can do this actually a lot more quickly than using the standard DNA FISH protocol as well. So we think that there's a really interesting future in the diagnostic direction. And we are also using it to look at brain human organoids. And we also get a really beautiful signal in a complex 3D culture. I think it will be very, really interesting to pursue this further in the future. So let me finish by acknowledging the people who have done the work. So Yang is one who developed very fast CRISPR. He was sick that morning, so he couldn't join a fishing trip in the Chesapeake Bay. And Raja is another member who worked with Yang. They really formed a formidable team. And then Yambo, I acknowledge, and Taylor, and many other members who are working on related projects in the lab. Let me acknowledge other collaborators, especially Bin Wu, assistant professor in biophysics and biophysical chemistry, who is an expert in live imaging. And we also work closely with Scott Bailey in the public health, who is an expert in biochemistry and structural biology of CRISPR proteins. Thank you very much.
Thank you, TJ, for an absolutely incredible talk today. I really appreciate you taking the time. I would like to first begin by inviting everyone to go into the chat, please. Please list your questions for TJ. We have about 11 minutes for a Q&A session before our next panel discussion begins. So I invite you all to please list them, and I will moderate the questions to Dr. Ha directly. So I will start. So TJ, thank you again so much for this. So I'm very curious, one of the challenges I believe I think that's agreed upon moving forward, right, is to be able to apply CRISPR, and particularly your new technology, in the setting of treating patients. And what we know in the biology is that there are certain cells that are the ones that need to be targeted. So I'm curious as to whether or not you've observed different dynamics of your fast CRISPR looking at multiple different cell types, and whether or not there's any differences based on either the nature of the cell that you're inducing the changes, or also potentially the state of differentiation. I wonder if you could comment on that.
Yes, so that's a really important question. So far we have only examined in detail the DNA repair process with CRISPR in standard culture cell lines. So these are immortalized and differentiated cell lines, and now we have made it work in induced pluripotent stem cells. So that will shine some light about differences between different cell types. We think that there are also great differences depending on the cellular stage. So in what cell cycle the cell is when we create the DNA damage, and also depending on where in the genome we are creating the DNA damage. So far, again, published work, we created DNA damage only in the genes that are transcribed. But what about genes that are not transcribed, or what about regions that are inactive, heterochromatins? And so now we have funding from NIH to study how different locations in the genome actually determine the DNA repair outcome. And again, because CRISPR-Cas9 can target anywhere you want, we should be able to begin to address these questions.
Very good, thanks. So let me just check for any additional questions right now. So then another question I have is there a possibility of sort of doing a multi-light excitation, right? So in other words, with the potential to do and hitting multiple genes, but using different light sources. Is that something that you've attempted, or are there technical obstacles to overcome to try to do that?
Yeah, so I think there are really exciting possibilities for multiplexing. And so let me first tell you about possible applications, and then tell you what is limiting us presently. The one reason why multiplexing is exciting is because, as you said, you can potentially make a cut at using different, let's say, different wavelengths of light. So you cut one gene and then wait for like 30 minutes and then cut another gene, and you can change the time delay between the two, and then study the relation between those. That'd be really great new tool. Another And that actually should be possible because there are different species of bacteria where Cas9 come from, and they bind different kinds of guide RNA. So you can actually do multiplexing there. But the limitation currently is that this Cajun group is usually activated more or less the same color. So you like to have an access to a Cajun group that can be uncaged at a much different wavelengths, and so that will be a major barrier in the future. But there are a lot of labs working on this, so there'll be a progress. Another I think reason why we went to the multiplexing is the following. So I've shown you results where people in the lab can activate and inactivate CRISPR using light, but these are two different experiments. So if you can activate a CRISPR using one wavelength and then only study the DNA damage response caused by the initial cut, and then use another wavelength of light to turn off CRISPR so that you don't have to worry about subsequent cleavage, that can be good for studying DNA damage response, but also really good for controlling genome editing. And so far, we have tried, but failed because if you put two modules in the same guide RNA, the perturbation seems to be too high to retain CRISPR activity.
Very good, thank you. So the questions are now coming in. I'm going to start. First comment, amazing work. Would the photoactivatable CRISPR only work on superficial part of tissue where light could reach when applied in the clinic?
So this is a very good point because we're using 400 nanometer light. So penetration depth is not as large as in the case of visible light or infrared. However, there are also people working in the photochemistry community developing Cajun groups that are uncaged using 2-photon excitation. So if you use, let's say, 800 nanometer laser from titanium sapphire laser, and then if you are able to uncage using 2-photon excitation, then you'll have a lot deeper penetration. And that would be the direction to pursue.
Great. The next question. You mentioned using the light control to prevent off-target gene cuts by CRISPR. Can you clarify how that might work?
Yeah, so here's the concept. Now it's generally agreed upon in the community that off-target effects are stronger if you have high concentrations of Cas9 inside a cell. And makes sense. You have more that can do the cutting, then more of the off-target cutting will occur. So in that context, because when you do imaging editing, so you only illuminate laser to a focus spot at a single green spot, then you are inactivating only a very tiny fraction of total Cas9 inside the nucleus. So active Cas9 concentration will be minuscule. It will be very, very small. So that will really eliminate off-target effects by itself, but also inactivated still caged Cas9 molecules are still there in the cell that also binds the potential of target sites. They will also prevent actually cleavage by the small number of activated enzymes. So that's I think that is the concept.
So there's a couple additional follow-up questions to that. I think there's a little bit of a concern related to how do you know that exactly where your light is shining, and sort of what the definition is of where the excitation is occurring, and to be able to obviously control to know that there's not off target. And wonder if you might be able to comment on that as well.
Yeah, that's a really good point. So in the demonstration that we made in a published study, we could actually know where to shine light on because we put dozens of GFP Cas9 near the cut site, just 30 kilobase pairs away from the cut site, by using the fact that we are close to a repetitive sequence. So that should apply for any site in the genome that is close to a repetitive sequence, which is very commonly seen inside the human genome. But there are also a lot of labs working very hard to develop live imaging technologies that can image any place in the genome using some Cas9 variants, even when it's not repetitive. And I'm sure some of them will succeed in the future because these are really brilliant young people. And then we can then use that to guide our uncaging light. So that is, I think, possible, at least under conditions where this kind of labeling is possible.
So we'll have time for just one last quick question and a comment. I think someone is also thinking creatively about, so is there a way to think about how to potentially automate this? So if there's a way to, photo activate, create a detector to sort of sense, the success of the activation, and then whether or not there's a way to actually potentially automate this in the future. So I wonder if you could speculate on that.
Yes, actually, this is exactly what Yang is planning to do. Instead of spending his time in the dark room and then doing it manually, you can basically imagine shining light onto a green spot and then see if the DNA damage response occurs. And presumably, if editing has been done, then you will no longer see additional DNA damage response when you shine the light again next time. So then you know that you're done, and then you can move on to another cell in the same sample. And you can automate this and edit a bunch of different cells and then analyze them using single cell sequencing.
Well, our time is up. Dr. Ha, thank you so much today for participating in our research retreat. And we can't thank you enough for this absolutely fantastic work and sharing this with us. Congratulations on this. And we really look forward to seeing the next stages of your success. Thank you so much for today.
Thank you so much, Nick.
So it's also my great privilege now to introduce Dr. Allison Moliterno, Associate Professor in the Department of Medicine and in the Division of Hematology, who will talk today and introduce the clinical applications of CRISPR technology, and will also moderate a lively panel discussion. So thank you, Allison, and I turn it over to you.
Thank you very much, Nick. And sorry, can everyone hear me? And see me. So it is really an honor today to present clinical applications of CRISPR. I'm from the Hematology Division at Johns Hopkins. And our research retreat theme, Transformative Technology, could not have been highlighted more by Dr. Ha's beautiful presentation and of what's coming up soon. But I'm going to take you to, but again, it's my great honor and pleasure to present clinical applications of CRISPR on behalf of the Department of Medicine Research Retreat and the committee that asked me to present this. So transformative technology in engineering and medicine couldn't be more apropos for what we just heard from Dr. T.J. Ha, and that's what I'm going to show you what's happening clinically. But a little bit of backup here. We want to cure sickle cell disease, and the NHLBI has a Cure Sickle Cell initiative initiated in 2018, and taken from their website, the goal is to accelerate genetic therapies, including gene therapy, clinical trials of new genetic therapies, including gene editing. And the time is right to push towards cures that are more accessible to the approximately 100,000 Americans and more than 20 million people worldwide who have sickle cell disease. Well, there's been a Cure Sickle Cell initiative for the past 100 years, really as long as we've had knowledge of the disease. The clinical description of sickle cell disease, of course, it's been around us for 7,000 years, but clinically identified in the early teens. And then really sickle cell disease has the first, the first molecular disease, right? It was the first amino acid sequence missense change identified in 1959. And then not long after that, hereditary persistence of hemoglobin F was identified. So really within just a few short years of knowledge of the DNA code, we find the molecular lesion and also information about hemoglobin F, hemoglobin regulation. And overlaid on this is the life expectancy of sickle cell disease, as we know, and the milestones that have occurred that have transformed the survival of sickle cell disease. Sickle cell disease and hemoglobinopathy really was also ushered in by acknowledgement of hereditary persistence of hemoglobin F and how this could modulate the sickle cell phenotype. And I show you here from March 1963, one of our hematology faculty, Sam Sharash, really brought this forward when he was studying hereditary persistence of fetal hemoglobin, which had been identified nearly 10 years before this in 1963. But as he was reporting these patients, there were 20 individuals in this story, in this report, that had SF. So they were S and F heterozygotes. And what they observed was that anemia does not appear to be a manifestation of this condition, and there's no evidence of a hemolytic disorder. So a natural experiment here really showed real mitigation of the sickle hemoglobin and phenotype. Dr. Sharash reported this in 1963, and nearly 30 years later, he capitalized on this observation, following this story for 30 years, and showed that pharmacologically you could increase hemoglobin F with hydroxyurea, and this markedly changed the natural history of the disease. I want to also mention that Dr. Sophie Lanscron, panelist, and myself were on the crisis review committee of the NSH study that was coordinated here in Baltimore through Johns Hopkins. And then finally, sickle cell disease, again, a perfect disorder to start gene therapy or consideration of gene therapy. We have so much knowledge of hemoglobin S and fetal hemoglobin. And again, in the early 1980s and up to 2000, gene therapy trials were starting to be initiated. And while not exactly applied to sickle cell disease, these were starting to really transform how we were going to treat genetic diseases. Unfortunately, this came to a halt in 1999 with the death of Jesse Gelsinger. But now I want to move back into the regulation of hemoglobin F because we have new opportunities, and the knowledge of hemoglobin switching is very important to bring forward to cure sickle cell disease and hemoglobinopathy. So we all remember from our hematology course, which we just are in the middle of for the medical school, that hemoglobin, the beta chain hemoglobin regulation varies during embryology and then fetal life, where we have different beta chains listed on chromosome 11 here that are turned on during embryogenesis and fetal life, and then get turned off once we're born, so that you start to bring the adult hemoglobin, which is the hemoglobin beta gene here. And you can see the relative contributions of hemoglobin. So we call this switch from turning off F and turning on beta the hemoglobin switch. So for instance, in sickle cell disease, you have a mutation in the beta gene here, S. As S is finally turned on, or the adult gene is turned on, you have increasing amounts of hemoglobin S, and your F starts to decrease. We know that children born with hemoglobin SS disease, they don't really start having phenotypic manifestations of illness until their hemoglobin F, the gamma chain, is really turned off. And so that we, again, have another very important piece of evidence that F is good to dilute the effects of S. And then I also want to show you beta zero thalassemia, which is going to be part of the trial that I'll discuss later. That again, you knock out the adult beta gene due to a promoter defect or a deletion. And again, your adult beta, it really can't come on because the gene is really dysregulated at this locus, and yet gamma turns off normally. So again, if we could fix this problem, let's keep the gamma genes on somehow and off the off switch, or keep gamma chains on. But there were barriers here because we didn't have complete knowledge of the fetal hemoglobin off switch. So while hereditary persistence of F was understood by all these gene deletions, we knew there was another locus here that was very important for really turning this switch off. And this finally was brought to light in 2008 and 2012, where the BCL11A enhancer in the gamma genes was shown to be the fetal hemoglobin off switch. So that was huge because now we can really consider how we're going to off the off switch. And we're going to do that potentially with the scissors here and with CRISPR-Cas9. So again, we could either scissor out the whole deletional region for the hereditary persistence of F, or we could go to the locus of the BCL11 erythroid-specific enhancer and disrupt that. And this is exactly what has been going on now in the clinic. So what listed here are the gene editing fetal hemoglobin trials. And I've boxed here the Vertex Pharmaceuticals trial that's using CRISPR to mediate the BCL11 enhancer disruption, so disrupt that. And it's applied to transfusion-dependent beta thal and sickle cell disease. And the trial started in 2018, just when the NIH decided that we're going to accelerate genetic therapies. And this is the outline of the trial. So what you have here is you have a patient with sickle cell disease or beta zero thalassemia. You're going to mobilize their stem cells. So again, take out the stem cells. Then you're going to electroporate them and then apply CRISPR-Cas9 to using the BCLA targeted erythroid enhancer disruption. This is our CRISPR-Cas9. Meanwhile, you have to go back to the patient and myeloablate their stem cells to give room for these, put these new edited stem cells back into the person. So a nice closed loop here. And we have now a brief report here just in December of 2020, coincident with our annual ASH meeting, where the first patients were reported. And on the left is the patient with beta thalassemia. Again, this is a person shown here on the y-axis, the hemoglobin in grams per deciliter, and the months after the infusion. Here's the infusion start date. Again, patients with beta zero thalassemia are transfusion dependent. So they have a lot of hemoglobin A, but this is all from transfusion. And they're anemic, so they need lots of transfusions even to keep a low hemoglobin. You can see now the emergence of the hemoglobin F in these blue bars, that many months after the single infusion, you can see their hemoglobins are normalized, and they have tons of hemoglobin F here making up their hemoglobin, and they don't need transfusions anymore. In sickle cell disease on the right here, we have the hemoglobin again and the percent of total hemoglobin in these boxes. Their hemoglobin S is in purple here. And again, we're not making hemoglobin S go away, but we're just turning on hemoglobin F. You can see again that hemoglobin F starts coming on, and they go from being anemic and mostly S, 71%, to really mildly anemic and 50-50 S and F. And this is sustained after several months. So the outcomes of this trial, their report was only a few patients, but at the annual meeting at ASH, they reported on, I think, 13 patients. So again, on the left, beta thalassemia, we can see all of these transfusions indicated by the dots for the 2.5 years before they had the trial. Here's the trial, and now you can see no more transfusions, and last transfusion right here by the little triangle. In sickle cell disease, we are looking for vaso-occlusive crisis, and also we're looking for transfusions related to sickle cell disease, because many of these patients require transfusions to avoid sickle cell crisis. And you can see vaso-occlusive crisis in red here, no more after the trial, and also last transfusions related to sickle cell disease, also no more after the trial. So really, really stunning. And now a lot of exclamation points, right? CRISPR-Cas9 gene editing is in hemoglobinopathy. It's safe and effective, and it's a success. This is an incredibly compressed timeline. We have identification of the BCL11A F off switch in 2008. CRISPR-Cas9 as a targeted disruptor, reported in 2012, and now clinical trial results in 2020. So are we talking cure here? And again, always we have concerns about the adequate knowledge of genetics. Do we have adequate knowledge of SF biology? And is this another disease? We have, of course, short-term and long-term problems to deal with, and CRISPR applications, of course, due to non-hemoglobin diseases. And now to help me with these questions, I'm really more than delighted to introduce my panel. And I'd first like to introduce Dr. Ada Hamash, the Frank Sutland Professor of the McKusick-Nathans Department of Genetic Medicine. She's the Clinical Director of Genetic Medicine and the Scientific Director of OMIN. I would also then like to introduce Dr. Sophie Lanscron, Professor of Medicine and Oncology, Director of the Sickle Cell Program for Adults at Johns Hopkins School of Medicine. And then finally, my Department of Medicine Research Retreat committee member, but also Associate Professor of Medicine and Pulmonary Division, the Johns Hopkins School of Medicine, Dr. Enid Neptune, whose specialty is basic science of lung disease and COPD. Let me just refer to my notes here. I want to really thank you all for joining this panel. We've got a lot to cover today. We had a little rehearsal session yesterday, and we're hoping that we can get to this panel, lots of discussion before the lunch break. All right, so Dr. Hamash, Ada, as the clinical director of the Department of Genetic Medicine and scientific director of OMIM, you've been the leading edge of both evaluating, identifying, and cataloging, and really communicating the knowledge of these at least 7,000 genetic disorders to patients and their families, and through OMIM, a tool that most of us use daily, basically to the world. So of all of us here today, you've personally witnessed the field of genetic medicine and gene therapy really explode, right, with all these technological advances. So the first question I want to ask you is really knowing thy gene, right? What is a good candidate for gene therapy? And certainly if we want to talk about CRISPR and Cas9, we can specifically address that. But what makes a genetic disorder either a great candidate for gene therapy approach, or perhaps not the best candidate? I want you to also with us express where the successes have occurred, and then also reverse real challenges. So Ada, could you take it from here?
Tiny little question you've asked me.
You got 3 minutes.
I need to apologize to everyone for the lack of a pretty background. It made me look like I was invisible and very scary, so I didn't want to apologize. So anyway, to start to answer your question. So first of all, you have to know the gene for the disorder. I mean, you can't target a gene if you don't know which gene causes what disorder. So we're up to 4,025 single genes identified to cause Mendelian conditions. That's today's number. Congratulations, everyone. So that's number one. Number two, it has to be, depending on what form of targeting you want to do, you have to know what the gene does, what cells it's expressed in, and you have to be able to get to those cells to target the gene, whether you're talking about CRISPR or any kind of gene therapy. And then you have to get it into the cells you want, into the cells, to have ongoing effects. So the death of Jesse Gelsinger in 1999 was absolutely tragic and brought the field to a dead stop. But it allowed for the important biology that needed to happen to actually understand what and how to perform gene therapy, what vectors are safe, which vectors target which cells, and what the stability is of the method. So you have to do that no matter what method you're using to get your gene into the body. You have to get it to the right tissues. So in terms of CRISPR, that's a whole nother layer of complexity. One is, so you can, if you obviously hematopoietic stem cells, you can get out and you can give back. So that's a wonderful system. Another one, what you don't want with CRISPR is you don't want to target germ cells, because you don't want to pass them on, and you don't really want to go too far beyond the organ that you want to use. So another place that's used, that can be targeted, has been targeted, hopefully will be targeted much more broadly, is the eye, which is a contained space and doesn't really communicate, the perfusion of it limits where those targets go beyond it. And you don't actually need to do a viral vector. You can just put it in the eye. So I think that answered the two questions a little.
Great question, great answers. So Dr. Landskron, Sophie, I'd like to turn to you. You are the director of the Adult Sickle Cell Disease Center for Adults at Johns Hopkins. You care for not only individual patients, but you also practice medicine at the population or disease level. Sickle cell disease, right, it's the first molecular disease, right? These gene therapies always want to go into hemoglobinopathy because we know so much about this disease, and these diseases are so morbid. And hematologists like us, we hate low hemoglobins, right? We want to fix hemoglobin, right? But there's also been a lot of pressure to move this quickly and to trust in the technology and our prior experience with gene therapy. We've seen some up and downs. So I would like you to really talk about the situation we're in. We got a lot of pressure from the federal government to cure sickle cell disease.
Yeah, well, right. So it's a really exciting time to be working in sickle cell disease. 1995 was when Dr. Swaraj published that paper, and it was FDA approved in 1998. We then went 19 years without another therapy for this disease, which is really unforgivable. And we know that there are major disparities in funding between sickle and some other single gene disorders, which we won't go into. There are now over 40 agents in the pipeline, and there have been 3 additional agents that have been approved. And hydroxyurea, right, the gene therapy turning on fetal hemoglobin, and we think that starting that in childhood probably is going to make a tremendous difference when patients get to see me in clinic as adults. The pressures to find a cure, right, clearly the NIH, Francis Collins, considers this a real goal. He eventually wants people to be able to just drive by, get their gene therapy and go, which we're a few years from. And people probably have seen the 60 Minutes where they gave a lentiglobin vector, right? The Bluebird trial where it was a lentiglobin vector gene. This was gene insertion, not CRISPR. And that led to a lot of excitement. And there was a lot of excitement. And one thing that they didn't realize, I went to clinic the day after the 60 Minutes, I thought it was pretty good, but they mentioned where this virus, where the viral vector comes from, and that it's HIV. And so, right, we have an obligation to really be able to explain this therapy to patients in a way that they can understand. There are so many ramifications of this. So currently, as of 2 weeks ago, the Bluebird trials were shut down. The NIH trials were shut down, and one other company shut down their trials because the Bluebird trial has had at least 2, if not 3 patients who've developed hematologic malignancies out of 40, which is obviously much higher than we see with other curative therapies like stem cell transplant. And so Bluebird actually just came out yesterday with their investigation, and they don't believe that this is insertional mutagenesis. They have taken out the leukemic cells and the gene inserted not in an oncogene. And so they really don't think that that's the case. And so the concern is, well, why did this happen to these 3 patients? And Allison, you showed a wonderful slide of the process, and one of those words in there was busulfan. And this goes back to, yes, we know a lot about the molecular diagnosis of the disease, but there's so much we don't know. And one of the key things we don't understand is the health of the stem cells in patients with sickle cell disease. And so that is a major concern. And so as we move towards other gene therapies and CRISPR, the CRISPR still requires the busulfan. And so this raises questions about the safety of that process. And then this other question of turning on fetal hemoglobin. So we know from the natural history studies, which were published 20 or 30 years ago, when you followed patients up for, 5 years or whatever, that yes, patients have much better outcomes if they have higher fetal hemoglobin levels. But it's a lot more complicated than that. We want the fetal hemoglobin to be present pancellularly, right? Because if it's not in the same cell as the sickle hemoglobin, it can't prevent the polymerization of the sickle hemoglobin. So if you don't have pancellular distribution of the fetal hemoglobin, it really doesn't help its helpfulness is limited by that. And so that's a key question. The other thing is that my colleague in the Bronx, Dr. Minnie, has published a series of patients who have elevated levels of fetal hemoglobin outside of the use of hydroxyurea. and they still have significant complications associated with their disease. They're probably just delayed. But certainly when you go to countries like our hospital in Saudi Arabia and Bahrain, where they have persistence of fetal hemoglobin, those hospitals are full of patients who are having pain associated with their sickle cell disease. So I think we're at the cusp of this, but we have a lot more to learn about how to do this well.
Well, thank you, Sophie, Dr. Landskron, for that really wonderful overview. We always brag about, that hemoglobin is the most important molecule, and it is. And hematology and red cells are the most important system. I mean, you need, but of course, Dr. Neptune, I will give you that you need a heart and potentially lungs to make it all worthwhile. We also brag that, well, we have stem cells and so we can manipulate these. And so you don't, Enid, right? It's not going to be a little bit difficult. But you have stem cells in the lung, but they're not so accessible. So Dr. Neptune, as a pulmonologist, I'd like you to sort of guide us through how could we use editing or gene therapy in an organ, right? You're standing in front of some lungs there. And how can we move this kind of technology into an organ system that may not be as accessible?
Oh, well, thank you to Allison and the whole panel and the retreat audience. The first thing I would like to emphasize is that sickle cell disease and thalassemias are the archetypal single gene disorders that should ideally respond to CRISPR-based technologies. So all of the concerns that Dr. Langstrom actually articulated are with the disorder that should best respond to this technology. But often cystic fibrosis is tethered to sickle cell disease as being a similarly attractive candidate genetic disease for CRISPR. And I want to explain why that's not quite the case. First, I want to clarify the skill set of sickle cell disease and why people consider this kind of the best disease for this technology. First is a hematopoietic disorder. The progenitor cell type for the affected cell compartment is very well known, can be isolated very efficiently from the blood, and can be subjected to gene editing or other genetic maneuvers. But most importantly, capacity can be created in the affected organ compartment, and the edited or corrected cells can be reintroduced into patients where they reliably repopulate the marrow and can correct the defect. An incredibly auspicious skill set. Many of you know cystic fibrosis is a rare recessive genetic disorder that's caused by mutations in the chloride channel CFTR that's expressed on airway epithelial cells. It's also highly morbid and associated with premature death, especially due to the extensive parenchymal lung involvement, as you see behind me. It's clear that replacing the mutant gene with the wild type gene in the affected cells would cure this disease. That's non-controversial, but especially if this is done before there is extensive lung damage. But the technical challenges are daunting and as of now, possibly prohibitive. In terms of ex vivo editing of progenitor cells, we do know the progenitor cells for airway epithelial cells, and that's due to really careful and meticulous work that's been done by several labs across the country and my research colleagues. We can actually, through a bronchoscopic approach, scrape these cells off the mucosa and actually survive them ex vivo. And organoid work that's been done with kind of airway epithelial cells suggests that we can actually use gene editing and other genetically targeting strategies in order to manipulate these cells. But what we cannot do is we cannot expand these cells. And more importantly, we cannot create capacity such that we could reintroduce these cells if in fact we could generate them in large numbers, such that they would go to the areas that are most affected and correct the disease impairment. So some of you would say, well, why don't you just deliver these CRISPR tools directly to the affected lung regions? And a challenge for lung delivery is that the lung is compartmental. And by that, I mean the proximal lung is distinct from the distal lung and is distinct from the airway. and the airspace. So what that means in each of these compartments, there's a specific kind of catalog of cell types that have their own unique skill sets. And there's also the problem of the huge area that's represented by all of these compartments. So targeted delivery for a therapeutic level of gene replacement is not yet achievable. So the bottom line is that to exploit the full potential of this amazing technology for lung disease, we have enormous technical challenges to address. And I asked my colleagues to consider whether similar challenges exist for your organ of interest. And that is the point where we start the discussion.
Oh, thank you, Dr. Neptune. That was amazing. I'd like to circle back also to Dr. Hamash, and also we have a few more minutes here to talk about the pace of change, right? All of us have been around a bit now in medicine, and we've seen, right, the introduction of gene therapy trials. We've worked with Dr. Sharash and Dr. Dover, our colleagues, and Dr. Mikusik, who really set the stage for genetic discovery and genetic medicine. But now we are moving very quickly. So Dr. Hamash, right, especially you cataloging OMIM, your practice, whole genome sequencing, I want to sort of clinical exomes, really how you keep up and how you handle the information and the pace.
So the pace is daunting. In 1989, jumping, chromosome walking and jumping found the CFTR gene. As I just told you, we have 4,000 recognized disease genes today. If we have 20,000 protein coding genes and 25,000 long non-coding RNAs and microRNAs that also have function, we have a whole lot more work to do to understand the role of all the genes in disease. And I believe most of them have a role. Maybe some in utero and you never see them. Maybe some are redundant, but most are going to have a role. We're moving quickly. It's becoming, for single gene Mendelian disorders, it's very difficult because many are described in very small numbers of patients, like one or two or a single family. And many are described in consanguineous families in which there are multiple affected individuals with multiple genes that have pathogenic variants. So sorting, particularly with description of a alleged new disease gene, it's very hard. So what do? We read a lot and we think a lot and we decide yes and no and wait for, we have piles of things where we're waiting for the next paper. But anyway, it's a dedicated group of incredibly hardworking people. In terms of the implications, so CRISPR has transformed science. It's our ability to do experiments, to answer a question in short order, whether in cells or in animal models, it's absolutely transformational. It's stunning. In terms of its implications for clinical care, I think there are places where it's already being used, and but I think we have to be very careful with what and who and how, both in terms of the technical challenges that Enid laid out so beautifully, but also, excuse me, in terms of the further implications if we want to make sure that CRISPR goes where it needs to go and doesn't go someplace else. And so there's a lot of work to do clinically, I think.
Thank you. Oh, thank you, Ada. Dr. Lansgren, I want to circle back to you because we're also, you're taking care of a population of patients and a vulnerable population, vulnerable on many levels. And we've all experienced this disruption of COVID and how a vaccine, an RNA vaccine, is now going to be pushed along into all of us. Hopefully all of us have been vaccinated much faster than it would have ever occurred, right? And yet we have to trust the system and encourage each other to take it and our patients too. So I want to bring this to you in terms of, well, when we talk about gene therapy for our patients with sickle cell, how do we protect them, but also provide the new technology?
Yeah, so a great question. And, the American Society of Hematology went around the country and asked patients and people living with sickle cell disease and their family and care members what was important to them. And the thing at the top of the list was cure. That was the most important, then pain, then fatigue, but really cure. So everybody does want a cure. But but people have also asked, what are you willing to give up? And one of the major concerns that people expressed in studies was the risk of developing cancer from either, at the time, I think most people thought that the use of busulfan as a myeloablative agent was going to be where that risk lay, but now we don't know. And so patients really describe not wanting to trade one disorder for another. Mm-hmm. And when you think about this so if the issue is aging stem cells, well, then maybe this is a therapy we want to do in children. I should point out that the youngest person who developed leukemia on that trial was 20. So that isn't necessarily the answer, but it's also this issue of what are you trading for? Are you trading for malignancy that's going to happen down the road? And, gene therapy, this is the disease for gene therapy when we know we can get it right. We are now just seeing a generation of children who started on hydroxyurea at a very young age who look so different than the population that I started taking care of, right? Their lives are much better. They have much fewer crises. They're missing school less. They're not having strokes. And that is giving them, 20 good years. Now, whether they'll get another good 20 years out of it, we don't know, because we still need to follow them. But I think we do need to bring the patient into this and really think about and make sure that they understand the consequences of this, and we have to be really cautious. And when the news came out about the Bluebird trial, there were 2 large community webinars to address concerns for the other 37 people who had this done to them. Their concerns about doing this, and the community concerns about doing this.
Oh, thank you, Dr. Lanskron. Dr. Neptune, I want to circle back to you because and Dr. Lanskron mentioned this, a disruptive change in therapy may subvert the need for gene therapy. So, and I'm going to go back to cystic fibrosis because again, we're always, we got to cure it with gene therapy, but wait, we've had huge disruption and benefit with small molecules and really transforming that disease, such that like hydroxyurea has changed the lives for children with sickle cell disease. They're not the same adults now, happily, with the same morbidity. And now we're seeing this transformation in CF with small drug therapy. So how do we balance this? Do we keep, well, I want you to discuss, but I also want you to discuss, do we leave all the investment in gene therapy when we have something else, or can we do both things at once? All right.
Okay. It's an incredibly provocative question. And I have to address the CF elephant in the room. And I think many of you and those in the audience are aware that we currently have effective small molecule drugs that address the CFTR impairment, not by replacing the mutant gene, but by correcting the functional deficits of the channel. Now, among those who are treated with these drugs, there are non-responders, and there are certain genetic genotypes that do not respond to these types of interventions. And that in itself could be a rationale for the exploration of gene replacement approaches such as CRISPR. But a more provocative discussion is whether the pursuit of CRISPR is maybe the most cost-effective approach for all patients with cystic fibrosis, given that it's likely, if successful, a one-and-done scenario versus the probability that patients will have to use a $200,000 to $300,000 a year drug for the rest of their lives. And one would hope that they start using these drugs early in life for maximal efficacy. Now, that's the type of delicate and very provocative discussion that's ongoing within the cystic fibrosis community, but can also extend to many other kind of single gene disorders where CRISPR is being considered. And it's certainly a very, very interesting discussion.
Oh, thank you, Dr. Neptune. Well, I have been given the word that we have 30 seconds left to the panel so that everyone can get to lunch. I want to thank my panelists. This was a really wonderful discussion. I want to let the world know that we met yesterday to try this panel out, and we talked for hours. So it has been wonderful that we've been able to get these points together in a timely fashion. and I look forward to seeing everyone in person. Thank you for coming to the panel, and please come back later for Dr. Doudna's address.
Thank you, Allison, Sophie, Ada, and Enid. That was absolutely wonderful. Thank you all for your time today. Really appreciate the clinical perspectives. So now we're going to go ahead and take a lunch break. We have about 30 minutes. So before we'll return at 12:50, Dr. Ha will begin his introduction at 12:50 of Dr. Doudna, who will give our keynote address. So thank you all for being here this morning, and we will see you shortly.
Good afternoon. My name is Taekji Pa. I'm professor of biophysics and bioengineering, biomedical engineering at Johns Hopkins University. I'm here to introduce our keynote speaker, Professor Jennifer Doudna from UC Berkeley. Because this retreat is held jointly between medicine and engineering, I really cannot think of any person who is better matched for the spirit and prestige of the occasion. Dr. Doudna won the Nobel Prize in Chemistry just a few months ago. We all celebrated the discovery of CRISPR-Cas9, used widely for gene editing. And her discoveries directly led to human clinical trials that are very successful, and also led to novel and powerful technologies to detect SARS-CoV-2, which is the causing agent for COVID-19 that we find ourselves in the middle of. Let me briefly tell you about her professional trajectory. She got her bachelor's degree from Pomona College in California. Then she went to Harvard University for her PhD and studied with Jack Szostak, who later on went on to win the Nobel Prize for his contributions in the discovery of the telomerase. After that, she moved to University of Colorado, Boulder, to do a postdoc training with another Nobel Prize winning scientist, Tom Cech, who had discovered RNA as a catalyst. She began her independent position at Yale University in 1994, where she stayed until 2002, when she moved to UC Berkeley, where she currently works. She won a number of awards, and I really don't have time to go over them. Let me highlight two of my personal favorites. One is L'Oreal UNESCO Award for Women in Scientists, and the other is award named after my favorite science writer and also distinguished scientist himself, E.O. Wilson, because he is really a big thinker, thinking about nature and science and humanity. And Jennifer has been really a strong advocate of ethical and equitable use of CRISPR technologies for improving human conditions. And she's also a great person for the audience, many coming from engineering, because she has authored, by my count, more than 50 approved patents, and also another few dozens that are soon to be approved. And she has trained so many amazing scientists as students and postdocs. And if you look at the list, it's almost like a who's who in RNA biology. And I recently learned one of her tricks. So when a student brings up a problem in a lab meeting and asks her, okay, what choice do I make? What do I do next? And she would just ask back, okay, what do you think? And that actually is a way to encourage students to think independently and become scientists on their own. I have my own recollection. So in 2003, I was in the middle of my third year as an assistant professor, and I came to NIH to serve on my first study section. As you may have done, it was a very nerve-wracking experience. I don't really recall anything I said. I don't even remember who else was in the room, but Jennifer later on came to me and gave me some compliments, encouraging me, and that really meant a lot. I'm sure her personal touch of this kind has also really helped a lot of the other young scientists. Okay, so I'm really delighted to hand over the podium to Jennifer.
Gosh, thank you so much, Takjuk, for this incredibly generous introduction. I didn't remember our exchange at the study section, but I know I have benefited from others who gave me similar kinds of encouragement. So I'm glad I could pass that along. Well, it's a wonderful honor for me to visit virtually at Johns Hopkins today, and I'm just thrilled to have a chance to speak with you about CRISPR technology and the future of medicine, given that we have this extraordinary tool for manipulating DNA. So let me kick things off here with my slides. And I thought I would basically break the talk into three parts today. I wanted to first speak about CRISPR at a sort of a high level, what it is and where it came from, how I got involved in it, because I'm not a genomics person and I'm not an engineer. So how did I get into the field? And I'll tell you briefly about that. Then I want to tell you a little bit about this technology for genome editing and where that's headed in terms of opportunities in science. And then I'll spend most of the rest of the talk talking about clinical use of CRISPR and how we think about this now that we have an extraordinary tool, not only for understanding functions of genes, but actually changing them, manipulating them, and doing that not only in laboratories, but in patients. So what's CRISPR? Well, it's a technology that allows programmable genome editing. And you're looking here at a picture of a CRISPR-Cas9 protein, the green molecule holding on to DNA. And the key to its action is that it uses a molecule of RNA, the yellow molecule, to determine where to hold on to DNA and where to cut it. And so this is a slide that is based on actual molecular structures of Cas9. And you can see that what it does is to unwind the DNA double helix and cut both strands of the helix at a place that's defined by the binding site of this RNA guide that the protein carries with it. So that looks kind of, I'm sure it might look kind of science fiction-y. It certainly did to me initially when we started thinking about the system. And, but I want to show you a quick video that illustrates CRISPR in action. So this is taken from a publication from a few years ago. And you're watching the Cas9 protein holding onto DNA, and there's the cut. So it actually functions like a molecular scissors to cut the DNA double helix. And that's the key to its activity, both in its natural host, which is in bacteria, as well as in other cells where it's used for genome editing. So where did this come from, and how does it really work? So this is in nature is a part of the system that bacteria use to fight viral infection. So this is a scanning micrograph of the surface of a bacterial cell in the process of being infected by phage. And these cells have very little time to defend themselves from phage before getting destroyed. So it's a very strong selective pressure for mechanisms that allow viral defense. And that's really what CRISPR is. It's an adaptive immune system that allows cells to protect themselves from future infection. And here's a quick animation of how this works in nature. So we're imagining a population of bacteria, some of which are getting infected by phage. And if these cells have a CRISPR sequence in the genome, they can acquire sequences from phage at this particular location. And those inserted phage sequences are flanked by repeated sequence elements. This is one of the ways that CRISPRs were initially identified bioinformatically. The cell makes an RNA copy of the CRISPR locus, and then that RNA transcript is chopped into shorter bits that each contain a sequence derived from a virus, shown in the yellow strands. That RNA combines with a second RNA called tracer and the protein Cas9 to form a surveillance complex that can search the cell looking for a DNA sequence, 20 base pairs in length, that matches the 20 nucleotide sequence in the guide RNA. And when that match occurs, the Cas9 protein cuts the DNA, triggering DNA degradation in bacteria. So in bacteria, this is a system that leads to quick destruction of an invading phage genetic material. So it's a great system for acquiring and then protecting cells from phage in real time. Now in the laboratory, so we began studying Cas9 with a collaborator, Emmanuel Charpentier, almost 10 years ago. And the question we had at the time was, how does this protein, which had been implicated genetically in the bacterial adaptive immunity pathway, how did it actually function on a molecular level? And that led to a fabulous collaboration with the Charpentier lab and our lab members, Martin Yinek and Chris Chylinski, who teamed up to do biochemical analysis of Cas9. And that data showed that Cas9 uses the two RNAs that I just mentioned to guide double-stranded DNA cutting. And the DNA cleavage is precise. Cas9 has two separate chemical centers that cut the double-stranded DNA. And Martin Yinek, doing sort of his classical, biochemical analysis of the system, was able to define the parts of these RNAs that are essential for function and create a single guide format for guiding Cas9's activity. So this created a simpler system than what nature had done by providing a single RNA molecule that included both the address label for the protein, the sequence here that could be easily altered, and the constant region on this end that provided the structural information for recognition by Cas9. And so for us, this work, when we did this original biochemical work, the goal of the project when we started was curiosity-driven research to understand this system. But as soon as we understood the way that Cas9 functions as an RNA-guided double-stranded DNA cleaver, it was clear that this would be a system that could be utilized in a different way in plant and animal and human cells, because of research that had gone on in the field of DNA repair and genome engineering for the previous 2 decades that showed that in eukaryotic cells, when double-stranded DNA cleavage occurs, cells, instead of degrading the DNA, typically will repair it. And they repair it by making a small change to the DNA sequence when the strands are religated, or by integrating a new piece of genetic material during homology-directed repair. And there were a number of terrific labs, including at Johns Hopkins, who had defined this and were studying technologies for introducing targeted double-stranded DNA breaks that would trigger this kind of repair and genome editing. The challenge was that those technologies relied on engineered proteins that were non-trivial to produce and required new protein engineering for each edit. And the great thing about CRISPR is that nature had come up with a way of programming a single protein with an interchangeable guide RNA that could be trivially altered in sequence to direct Cas9 to any desired place in the genome. And so this was our proposal back in when we published our work originally with Emmanuel in 2012, was to show how this could in fact work as a simple programmable technology for genome editing. And so I wanted to show you one more quick video here that imagines how this works in a eukaryotic cell. So the Cas9 protein with its guide RNA can enter the nucleus either with a nuclear localization signal or during cell division when the nuclear envelope breaks down. And the protein is able to search the entire genome to find a sequence that matches the sequence of the guide RNA. We know that the DNA then unwinds to form an RNA DNA hybrid in the Cas9 protein, and that triggers targeted DNA cutting. Now, in eukaryotic cells, that cleavage event is, rather than triggering DNA destruction, that cleavage is recognized by repair enzymes that actually carry out the editing. So when we talk about CRISPR as a genome editing technology, it's important to appreciate that it initiates the editing, but what actually carries out the changes to DNA are the events that happen after CRISPR-Cas9 does its work, at least in its native format. And I'll tell you later briefly in the talk today about a way to use Cas9 for more direct chemical modification of DNA. So this, really has this sort of certainly redirected the focus of my lab, and many other groups around the world quickly began using CRISPR as a technology for genome editing. And it's clear that the possibilities are really large, because we have now a technology that allows not only fundamental discoveries to be made more quickly about gene functions, but also real opportunities to use genome editing in the clinic and for agricultural applications that are going to provide, I think, practical solutions to genetic disease and to challenges of things like climate change in the near future. So it is a really exciting time with this technology coming together with, of course, a number of other advancements in both biological research, clinical medicine, and computing that are converging to provide just really extraordinary opportunities. I wanted in particular to mention that this is not just a fantasy about using CRISPR in clinical medicine, but it's actually now part of ongoing clinical trials. And one of the trials that's really attracted a lot of attention is work that's being done to treat patients that suffer from sickle cell disease. So this is a very well-known genetic disorder resulting from a mutation, a single base pair mutation in the human beta globin gene that gives rise for people that inherit 2 copies of this gene to a mutated form of hemoglobin that is prone to aggregation and gives rise to the classic sickle shape of blood cells that contain it, and leads to all of the downstream effects of this disease that include organ damage and terrible cycles of crises that are difficult to manage clinically. And the amazing thing about CRISPR is that it's a technology that in principle allows a mutation of this type to either be directly corrected or for a change to be made in another gene, such as fetal hemoglobin, that can suppress the effect of the original mutation. And this has been now the subject of multiple clinical trials whose results have been announced, including some new data that just got announced today. And I just wanted to point out that this is a picture of Victoria Gray, who was the first US sickle cell patient to receive CRISPR as therapy. And she's made headlines over the last year or year and a half because the therapy has worked well for her so far. So she's been effectively cured of the effects of her disease through the activity of CRISPR. And this has been clearly a real watershed moment in the field of gene therapy, and I think gives all of us hope that there are real opportunities in the future for people that suffer from genetic diseases of this type. Of course, a big challenge is one of the challenges with this, of course, in addition to all of the considerations in a typical clinical trial, I think is going to be ensuring that the technology is affordable and accessible in the future to patients that need it. And so that's something I've been thinking about and working on a lot with colleagues at the Innovative Genomics Institute in the Bay Area that is a partnership between Berkeley and UCSF. And I might say a little bit more about that at the end of the talk. I also wanted to point out that it's been increasingly clear that we as a scientific and clinical community, we need to be vigilant about the use of genome editing in different systems as the technology continues to advance. And for the past several years, one topic in particular has been the use of CRISPR in the human germline to make heritable changes that in principle could allow changes to be made that would be passed on to future generations of humans. Really a kind of a profound thought. And I've been part of a number of international groups that have been looking at this as an ethical and societal challenge. And there have been now a number of reports, including this one shown here, that have been released in which international teams of scientists have looked into the technology and made recommendations for how it can be used ethically going forward. I don't think this issue is going away. I do think that it will require continued discussion and transparency as the technology advances. So I wanted to now turn to a little bit more on the science side of things and talk to you in particular about two areas where I see really interesting opportunities for discovering fundamental mechanisms that could help us advance CRISPR as a therapeutic that will be both safe and effective and eventually widely available and affordable. So those are big challenges. And you know, how do I think about that as a biochemist and somebody who works on molecules and mechanisms? And so I just wanted to really point out talk briefly about two areas where we've been working on the mechanisms of CRISPR and how to deliver it to be able to eventually have a technology that will be as effective as possible. And one of the first question I want to pose briefly is how do we ensure the precision of editing? Because as I mentioned, CRISPR is a molecular scissors that cuts DNA in its native form, but it hands over the actual editing chemistry to the cell. And so that poses a challenge for people using it because we have to figure out how we control the way that editing is done. And so one of the ideas that has emerged over the last few years, largely initially from the work of Alexis Kumar, working with David Liu in Boston and in the lab of Professor Kondo in Japan, both realized that CRISPR offered an interesting opportunity to directly correct disease-causing mutations, such as single nucleotide polymorphisms or SNPs, that are known to cause disease. And this is just a pie chart from one of the David Liu publications showing that if, for example, one could correct AT base pairs to GC pairs and to correct an AT SNP mutation by a process known as A-base editing or ABE, that in principle, one would then have a tool that could fix the pathogenic mutations responsible for about 48% of the known pathogenic SNPs in humans. Kind of an amazing thing to think about. So then the question is, well, how would we do that with something like CRISPR? And so what these laboratories did was to ask whether they could take advantage of the natural ability of Cas9 to unwind DNA and expose part of the DNA as a single strand to use this as a way to get targeted editing of nucleotides that were briefly exposed in this kind of a fashion by Cas9. And so in a strategy, this is an illustration from a paper published by the Liu Lab. The idea here was to append to the Cas9 protein an editing domain, an activity that would be able to make that kind of chemical modification, in this case, of an A and adenosine nucleotide from an A to an inosine, which could then be corrected to a guanosine during DNA replication in cells. So kind of a nifty idea. The problem was in nature, there aren't any known editing proteins that do this kind of chemistry. In other words, there aren't known editing functions that act on single-stranded DNA to convert an A to an I to a G. However, in bacteria, there are proteins that do this, and they act on transfer RNA or tRNA. So they're involved in editing tRNA, such as this protein shown here, known as TAD-A. So TAD-A is a dimeric protein, and its function had been very well studied and characterized. It recognizes the anticodon loop of tRNA and converts A to I in tRNA. And so the strategy taken by the Liu and Kondo labs then was to use directed evolution to evolve the natural activity of a tRNA base editor to be capable of DNA base editing. And so they went through a number of rounds of selection. And the general strategy is shown down here, where the Cas9 protein was appended to the editing function. Sorry about that. And these had, these were sort of different versions of this chimeric protein that were evolved over time in the laboratory and had different degrees of functionality, primarily assessed by analyzing base editing in a cellular assay, so not really looking directly at the function of these proteins per se. So that's where we got involved. So we started working with David Liu's lab a few years ago to do what initially seemed like it might be fairly pedestrian work to just characterize the biochemical activities of the base editors. And fairly quickly, we found something quite extraordinary, which was that most of the base editors, the earlier versions, had very, very low editing activity measured in a biochemical assay, shown on a graph here, where we had to do typically days-long incubations to see substantial base editing. But Audrey Lapinate, the postdoctoral scholar who was doing this work in our lab in collaboration with the Liu lab, tested at the time the most recent version of the A base editor out of the Liu lab called ABE8E, and found that it had incredibly fast editing kinetics, especially relative to these earlier A base editor proteins. So we scratched our heads and said, what's going on there? That must be something very special about this newly evolved A base editor. And so in order to figure out how it might work and what might be special about this protein, we partnered with the lab of Pete Beal at UC Davis and his student Cody Palumbo to ask whether we could trap this fast editor in the act of editing DNA. And so the strategy here was to use an approach that the Beal lab had developed for studying TAD-A in its native function as a tRNA editing enzyme, in which they had shown that you could substitute the A nucleotide that was being edited by the protein with a nucleotide analog called 8-azonebularin that literally captures the chemistry of this reaction in midstream by forming an analog intermediate shown over here that cannot be processed further. And so very painstakingly, Audrey and Cody made substitutions along the length of the DNA. We were trying to figure out from earlier structures where we might get the best exposure in the context of this R-loop structure that forms in Cas9. And eventually, we were able to trap a structure and visualize it using cryo-electron microscopy. And this involved the great work also of Gavin Knott, another postdoc in the lab. So Audrey and Gavin teamed up to do the structural biology of this complex. And I just want to quickly show you a little video of the structure. So the white part of the molecule is the Cas9 protein. In red and pink are the editing parts of this chimeric protein. And hopefully you can see that the DNA, here's the DNA double helix, entering the protein, entering Cas9, and the strand of DNA that is unwound and melted out and not associated with the guide RNA, which is buried inside the protein, is in fact exposed to the editor and is bound in the active site. And what was very exciting to see was that the 8-azonebularin substitution is in fact buried right inside the active site. And when we looked at where mutations occur in this TAD-A evolved protein that stabilize the structure, we found that they largely lie in this binding pocket here that enables the DNA to deviate into the active site and be recognized accurately by the protein. And in a lot of other data that I won't show you, so we were able to show that this is a very fast editor. In some ways, it's too fast because it actually allows the protein to, as Cas9 carries out its natural function of scanning along DNA, looking for possible binding sites that are complementary to the guide RNA, we know that it transiently melts a few base pairs of DNA, and that allows this very fast base editor to actually get in there and do some editing. So if you look along the length of the DNA, you do find that there are stochastic events that are editing events that are happening along the length of the DNA in the process of getting to an actual bona fide binding site for Cas9. So I think ultimately the best base editor is probably going to be a sort of a Goldilocks solution where we find an editor that's fast but not too fast, so that it correlates better with the kinetic behavior of this search mechanism of Cas9, so that you have an editor that is truly dependent on full formation of an actual R-loop structure in the DNA. And that's something that multiple labs are now working to do. I also want to briefly talk about the other, what I think is another sort of big challenge in the field, in addition to editing precision, is editing delivery. How do we ensure that editing molecules are introduced into the right cells, especially if we're doing this for clinical use in a patient? Of course, one can do ex vivo editing and edit cells outside of the body, but ultimately it will be, I think, very valuable to have delivery vehicles that allow in situ editing in the patient without having to do ex vivo editing in the lab. And just to highlight this, I wanted to point out that in work that was published last year from Carl June's laboratory, where they were using CRISPR to engineer T cells in patients with refractory cancer, this paper showed that the technology for ex vivo editing is quite robust and also seems to be safe in these patients. And the idea is that T cells are harvested, they're edited in the lab, the editing is validated, and then the cells are returned to the patient. And so although this technology works, of course, it is time consuming, it's expensive, and it poses challenges for expanding the technology to all of those that might benefit from it. And so in our lab, we've been thinking a lot about how we might approach this question of delivery. And this has really been spearheaded by Jenny Hamilton, a postdoc who came to the lab with from a virology background and really wanted to think about this from the standpoint of how we take what we know about the way viruses infect cells selectively and apply that to delivery of CRISPR molecules. And she's now working with a team that includes Connor Sushita, a bioengineering graduate student, and Abby Stahl, another very talented postdoctoral scholar in the lab. And so what they've been doing together is using various types of virus-like particles, or VLPs, to deliver Cas9 as a pre-assembled protein with its guide RNA. And so the strategy here, this is different from using a viral vector for delivery where Cas9 might be encoded genetically. Here what we're doing is gutting the virus genome of, in this case, an HIV type virus, a lentivirus, of the genes necessary for viral replication, but retaining those necessary for forming viral capsids. And by making a fusion protein of the GAG polypeptide with Cas9, we can conduct plasmid transfections of these 4 plasmids and into cells, and then collect supernatant that contains assembled capsids that look like this, where Cas9 is appended to the viral capsid. It can be cleaved off with a protease. And importantly, we also can include a protein, a viral sequence here that encodes proteins that we might want to have integrated into the gene edited cells during the delivery of this type of viral capsid carrying the Cas9 with its guide RNA. And I'll just show you one example of how we're using this. So you know that probably many folks here are familiar that HIV naturally infects T cells and relies on recognition of receptor molecules on the surface of those cells. And so what Jenny, Connor, and their colleagues at UCSF, David and Brian, who are both clinical fellows in the lab of Alex Larson at UCSF, these folks have been working together to ask whether they could target specific kinds of T cells in a mixed cellular population using viral-like particle delivery. And we're very excited that this strategy seems to be working. So this is just one set of data showing that we can take a mixed cell population and use virus-like particles that are specific for CD4 positive cells and get editing of those cells without editing of other types of T cells, such as CD8 positive cells. And so this is a strategy that we hope eventually will be useful for delivery in vivo. And we're now working in a mouse model to see if we can get effective delivery by providing these kinds of virus-like particles as an intravenous injection to get to observe the kind of editing that previously would have been done and observed only in ex vivo types of experiments. And that's about where we were when this virus appeared on the scene about a year ago, at least here in the US, that we began to, I think many of us realized that, we were in really deep trouble. This is an RNA virus and has caused many of us to kind of, rethink, at least in the short term, and in some cases, maybe in the longer term, of how we're going to direct the focus of our research efforts. And in my own case, this made us go back to work that we had been doing a few years ago and then had kind of passed off to others, which was taking advantage of the wide diversity of CRISPR-Cas proteins that occurs in nature. So these are as part of bacterial immune systems. They've diversified extensively, probably in response to viruses. And so we've been over the years with initially Emmanuel Charpentier and then with Jill Banfield at Berkeley, looking at different types of CRISPR-Cas systems. And one of the classes of proteins that we and others have investigated are proteins called CRISPR-Cas13. And these are enzymes that have the similar ability to recognize nucleic acids with an RNA guide, but in this case, they recognize RNA rather than DNA. And furthermore, as in some work that was shown originally by Alexandra East-Seletsky, we could take advantage of the natural function of these CRISPR-Cas13 proteins, which is to cleave RNA non-specifically upon binding to a target sequence. And by using reporter molecules labeled with a pair of dyes that are initially quenched, fluorescent, are not fluorescent because they're quenched, they're very close in proximity. When an activated Cas13 cleaves this type of reporter, then a fluorescent signal is released and is detectable. And so this was an idea that we published back in 2016 that's now been expanded by many others and is being utilized as a detection system. So starting about a year ago, David Savage, Patrick Hsu, Melanie Ott, and Dan Fletcher and I, 5 professors in the Bay Area, formed an academic consortium to try to accelerate this work and see if we could come up with an actual point of care test that would use this kind of approach for direct RNA detection. And so Melanie Ott has published a recent paper that showed kind of an initial success with this strategy. And we're now in the process of developing it further and taking advantage of this fundamental chemistry by accelerating the rate at which this fluorescent molecule release occurs using a tandem CRISPR approach. And I'll just very briefly mention that we've also partnered with a couple of local companies to develop a simple microfluidic chip that would allow the chemistry of CRISPR to be used in this point of care setting with a detection system that was developed by the lab of Dan Fletcher. So it's been a really fun, really exciting opportunity to bring together the virology and immunology expertise of Melanie Ott and her colleagues at the Gladstone Institutes bioengineering capabilities of Patrick Hsu and Dan Fletcher with the CRISPR work that's been going on in a number of laboratories, including ours over the years. We're cautiously optimistic that we're going to actually have this point of care device running here in the next few weeks, and we'll be doing a study at Berkeley with our students to see how well it works as a surveillance test for monitoring infection in asymptomatic individuals. So in summary, we've been continuing to work on understanding the mechanisms of genome editing proteins. I think that my experience over the years has always been that the more you understand about the molecules and the way they work, the better you can use them in either their natural settings or engineer them to do new things. And so we continue to take that approach in the genome editing arena as well. I'm very excited about the possibilities of new kinds of genome editors that are quite small and might be better for delivery into cells. And of course, we're excited about the future potential to achieve in situ genome editing with new delivery technologies, such as the viral-like particle approach that I mentioned. And I'll just thank an incredible team. So we have a great group here where this is taken right outside the Innovative Genomics Institute building at Berkeley. I've had just extraordinary students over the years. It's just such an honor to work with people that are so talented. I want to thank all of the folks mentioned here some of them I mentioned specifically in the talk, but all of them have been involved in especially the recent efforts with CRISPR diagnostics. And then of course, longtime collaborator Jill Banfield and Emmanuel Charpentier, with whom we started the whole effort to study Cas9 in the first place. And then I have to also give a big shout out to our funding agencies that make all of this possible with their generous support. Thanks very much, and I'll be happy to answer your questions if you have them.
Thank you, Dr. Downa. This was absolutely an amazing experience for all of us. We cannot thank you enough for your time today and this wonderful presentation that covered many topics. Since we have just a few moments, I'm going to jump right to the questions. And the first one is, thank you for this eye-opening talk. For in vivo lentiviral CRISPR editing of T cells, are you concerned about immune response and clearance to bacterial origin proteins, for example, Cas?
Yes, thank you for that question, Emily. We are concerned about that, and that's been the focus of the work of Abby Stahl in particular. So we're doing a lot of comparative studies right now of the immune response in mice to these virus-like particles compared to other modes of delivery, in particular using a viral encoding mechanism like AAV. And so far, we're actually quite encouraged that these viral-like particles are certainly no worse than AAV, and in some cases seem to be better tolerated in these animals.
So I'm seeing on my cameras here. TJ, did you have a question?
Yes, I had a question regarding the diagnostics. So how so usually the Cas13 based detection requires this bystander effect. That it's cutting other RNA lines specifically to generate a signal, and but also it requires preamplification using a LAMP, which can take some time and also requires a high temperature treatment. Is there a way to skip a LAMP so you can use a CRISPR system, for the whole process of adding patient sample and getting the signal out without having to preamplify the nucleic acids?
Yeah, that's a great question, TJ, and thanks for bringing that up, because I want to make that clear that that's really been our approach in this consortium, was to ask exactly that question, because one can certainly make lots of copies of viral RNA for detection, but then, as you said, it takes time. You have to go through typically steps of converting the RNA to DNA and then amplifying the DNA, and then if you're doing RNA detection, converting it back into RNA, which all takes time and heating and various things that are going to be more complicated, especially for point of care use. So we asked, could we just use CRISPR-Cas13 in its natural fashion of, just as a direct RNA detector? Would it be even possible to get the kind of sensitivity that, and accuracy that one would need for a good test? And this was this was kind of the original work that was led by Melanie Ott and her team that, they published this paper just a few months ago showing that the answer is yes. So certainly Cas13 provides the kind of sensitivity and accuracy that you would need. The challenge is that it's a bit slow, right? It's a bit slower than you would like in terms of, especially as you get down to very low amounts of the target RNA that you're detecting. And that's where we've been focusing our efforts more recently. So I didn't get into the details here, but in the work of Tina Liu, a wonderful postdoc in the lab. So she's really led a group of people as part of this consortium to couple Cas13 with a second CRISPR nuclease. It's a general nuclease, but we've set it up so that when Cas13 recognizes a target RNA and starts to cleave other molecules, what it cleaves initially is an activator for the second nuclease. And we figure out a way to chemically stabilize that activator. So once it gets turned on, it's on. And then that nuclease just takes off and releases a signal. And there's a lot of details in there, but I think, we're feeling pretty optimistic that this is going to provide a very fast way of detecting RNA directly without requiring any kind of pre-amplification through a DNA intermediate, like you mentioned.
And we have a comment from Dr. Cass, who says, with 3.2 billion bases, 160 million unique 20 base sequences, what is our understanding of how the kinetics of this happens? CRISPR is wandering about with a guide and samples all of these fast enough. Clearly it works, but seems very remarkable kinetics.
Yeah, absolutely. That is, you put your finger on one of the real head scratchers about CRISPR that I think is still under investigation is how does it find targets fast enough to be effective? And there are now several studies, including one from us with one of our collaborators at Berkeley, Robert Tejan, using live cell imaging to look at the search mechanism of CRISPR-Cas, and other people have looked at this as well. And I think part of the answer is that this is a protein that has very rapid binding kinetics to DNA. So it's rapidly binding and releasing DNA. There's also some single molecule evidence. I think this is partly from your work, TJ, that suggests that the protein has an ability to process along very short lengths of DNA. It doesn't seem to be processing along long stretches of DNA, which is again maybe consistent with this idea of very rapid search and release, kind of binding and release kinetics. But the bottom line is that probably in reality, one really needs to have lots of molecules of CRISPR-Cas9 in any particular cell that's getting edited, so that you have a reasonable chance and a reasonable timeline of one of those molecules actually finding its target.
So I have a question myself, and my question is basically geared towards, while it's been shown to obviously function in various different cell types, my question is whether or not even within the same cell type that has the potential, the ability to flux in terms of the state of its differentiation, right? Of whether or not that you or anyone else has observed any kind of change in either the kinetics or the efficacy of the CRISPR sort of based on the dynamics of a cell and the state it exists at the time of editing.
Yeah, the answer is yes. And there does seem to be quite a bit of variability, depending on the type of cell, certainly, and as you implied, the state of the cell, whether it's whether it's actively dividing or not, the rules for that are still, I would say, not well defined. It's probably going to, certainly the editing part of the process, not just the Cas9 initial searching of DNA, but the actual editing events will rely, I think, a lot on what's happening in the cell, the type of cell, the way that DNA repair normally occurs in that particular type of cell. And there's lots of evidence for that now. So I do think that one of the real challenges in the future in the field is to better understand that and to be able to control the way the editing works. And you know, that still is, I would say, very much at the forefront, sort of cutting edge of the field, is to figure out how to control editing that happens with these proteins.
Maybe I have one. Is there time for another question?
Quickly.
Oh, sorry. So it's not actually about the research itself, but you know, there are a lot of young students tuning in today, Jennifer, and I know, but not many people know that you actually briefly took a leave from your professor position to join pharmaceutical biotech company for about a month, and you came back, and then you discovered CRISPR-Cas9 activities. So can you say maybe a few words about you know, the importance of basic science research, and maybe, you know, really encouraging the young students to think more about that, but based on your own experiences?
Sure. Well, I'll be brief, because I know we're almost at the end of the time here. But yes, you allude to the fact that back in 2009, I briefly took a leave of absence from Berkeley, where I'm a faculty member, and joined Genentech. And my thinking at the time was, you know, I was sort of at a point in my career where I thought, you know, I've had a lot of fun with my science, but I wondered, you know, am I really doing work that's going to have an impact? You know, and I had this opportunity to go to the company, and I thought it might be, you know, a really exciting way to take my research in a more focused direction. However, I have to be honest that when I got there, and it's a wonderful company with a lot of really fantastic people, including folks who had left academic positions to join Genentech. But for me, I just, you know, I really missed the intellectual environment that I was used to at Berkeley, and just the kind of free thinking, you know, opportunities to pursue science in directions that seemed interesting. And as you said, TJ, I then, fortunately, Berkeley took me back, and I went back and I really did refocus my efforts on CRISPR, just feeling that, you know, I really felt there was a kind of a nugget there that I wanted to unravel. And I couldn't have predicted where it ended up, but, you know, I think it does speak to the serendipity of science, and also the importance of pursuing work that you really feel passionate about.
Thank you.
Okay, so again, Dr. Donna, thank you so much for being here today. Thank you for participating as our keynote speaker. It's been a great privilege for all of us to have you here and to hear about all of your great work. We wish you all continued success on behalf of myself, the chairs and the deans here, and all of us at Johns Hopkins. Thank you so much for everything, and we look forward to seeing many great things from you in the future.
Thank you very much for hosting me. It's been a real pleasure.
Okay, everyone, this will conclude our day one session. Hope you all enjoyed everything today. Please join us again tomorrow morning at 8:15. Dr. Anderson will give his opening remarks and his review of the last year. And we will continue and look at all of the achievements that have been done tomorrow from everyone here at Johns Hopkins, from our trainees to our junior faculty. So again, thank you all for participating today, and we'll look forward to seeing everyone tomorrow morning bright and early. Thank you.